Good morning or afternoon to everyone, depending on where you are today, and welcome to the Critical Path Institute, or C-Path's Transplant Therapeutics Consortium TTC Workshop. The workshop is titled Accelerating Medical Product Development in Kidney Transplantation Through a Public-Private Partnership. And we are, number one, very grateful just for you taking the time to be with us today. Up front, I'm going to do some housekeeping items. My name is Nicole Spear, by the way. I'm the Senior Project Manager for TTC. I've been with the consortium since its inception, pretty much. One thing to note is feel free to take breaks as needed. We've got 4 hours on the books and we have a lot of content to get through. So feel free just to kind of carve out that time for yourself. We don't have any breaks built into this. Again, some housekeeping things. The audience members are going to be muted throughout the workshop. If there's any questions, just feel free to type them into the Q&A feature that you can see through this portal. A recording of the workshop and the slide deck will be sent following this, as well as a summary. Finally, and most importantly, just note the speakers have pre-recorded sessions. So when the sessions are complete, we'll follow that with a live panel discussion where, again, questions will be addressed that you can enter into the feature. In a moment, I'm going to be handing this presentation off to Özlem Balayan, who is the Deputy Director for the Division of Rheumatology and Transplantation Medicine at FDA, and she also serves as the FDA liaison to the TTC itself. Dr. Amanda Klein, who serves as the Executive Director of the TTC, will also be preparing some opening remarks just to kind of set the stage for the day. With that, let me just quickly walk through the agenda. So session one is going to be focusing on what C-Path TTC has learned through the process of working with regulatory authorities to qualify a composite surrogate for a long-term graft loss after kidney transplantation. It will be led by Amanda Klein, again, the Executive Director, Luke Kaczynski, who is a quantitative medicine scientist at C-Path. And then the panel discussion is going to be moderated by Bill Fitzsimmons, who is a lead scientific advisor with the TTC. Session two is going to dive into the use of real-world evidence data in transplant clinical trials. And our speakers are going to be Luke Kaczynski again, Alice Toll, who is a biostatistician at the United Network for Organ Sharing, which is also called UNOS. Dr. John Concato, who is the Associate Director for Real-World Evidence Analytics within CEDAR at FDA. And then the panel discussion will be moderated by Amanda Klein. Session three will be focusing on where we go in terms of potential future endpoints in kidney transplantation with speakers from Dr. Peter Nickerson, a professor of internal medicine and immunology at the University of Manitoba, Dr. Bill Fitzsimmons, who I previously introduced, Rebecca Speck, Dr. Speck, who's a clinical outcome assessment scientist with C-Path, and then Dr. Dave Tabor, who is with the University of South Carolina. The panel discussion will be moderated by Dr. Mark Stegall, who is joining us from Mayo Clinic and who also serves as a work group chair for the TTC. And then the closing remarks will be led by Amanda Klein, who, again, is the Executive Director, Kenneth Newell, who's a representative from American Society of Transplantation, and Ulfmeier Krisch, who is the CSO at Veloxis, and also both serve as work group co-chairs.
Okay. Hello, everybody. I'm Amanda Klein, the Executive Director for the Transplant Therapeutics Consortium. I'm very grateful to be here today to lead this workshop. This has been years in the making, and it's been through collaboration, commitment, dedication, alignment, and incorporating the entire transplant community. So a little bit about the Transplant Therapeutics Consortium. The TTC was founded in 2017 to accelerate the medical product development process for transplantation. And this is accomplished by convening stakeholders from diverse settings, including industry, clinical settings, academia, and regulatory agencies. The current unmet need is developing novel immunosuppressive therapies in transplantation that improve long-term graft survival. And so our current efforts are geared towards long-term graft survival, specifically in kidney transplantation. So as part of the Critical Path Institute, we lead these public-private partnerships in which we carry the regulatory expertise. And we have Ozan Boleyn. We're grateful for her leadership and being a champion and dedication as part of the TTC. But you can see here that we have diverse stakeholders that have been part of this this journey. Next slide. So really, without the commitment of the entire transplant community part of the TTC, you can see we have current C-Path staff and former staff, and then advisors. But this regulatory endorsement has been supported through funds from TTC members, but also through a broad agency announcement contract with FDA. So I want to thank everyone, and I also want to thank a special thank you to Inisha Doherty, who was the previous executive director of the Transplant Therapeutics Consortium from inception.
Right. So graft failure has significant negative outcomes, including return to dialysis, decreased quality of life, retransplantation, and decreased donor pool. Graft survival is the most important outcome for people living with a kidney transplant. Currently, therapies have drastically improved short-term outcomes, with 1-year graft survival hovering around 91%. But the challenge for sponsors is executing registration trials of a feasible size to support superiority claims for 1 to 2 year studies. When we look at long-term graft survival, this remains suboptimal. 5-year survival rates in primary kidney transplants in the United States is around 79%. And so the challenge for sponsors is designing practical trials with a 5-year primary outcome. The currently accepted endpoints are not sufficient to assess novel immunosuppressive therapies aimed at improving long-term graft survival. Moreover, given that the causes of late graft failure are multifactorial, predicting such failure accurately with a single marker may not be optimal. A critical path innovation meeting was held in which FDA expressed agreement on the unmet need in transplantation and supported the qualification of a composite score as a reasonably likely surrogate endpoint for use in support of drug approval through accelerated approval pathways. Thus, this talk will focus on the regulatory qualification procedures and the qualification status of the Ibox scoring system as a reasonably likely surrogate endpoint with FDA. The Transplant Therapeutics Consortium, the TTC, has been working to qualify a composite biomarker with FDA and the European Medicines Agency, EMA. Qualification is a formal process of regulatory review and acceptance. It is a conclusion that within a stated context of use, the biomarker can be relied upon to have a specific interpretation and application in drug development and regulatory review. As shown on this slide, FDA and EMA have separate guidance documents for applicants seeking qualification. Once qualified, a biomarker becomes publicly available for use under the specified context of use to streamline drug development and review of regulatory applications, providing the confidence for both the submitter and the reviewer. As part of the 21st Century Cures Act, the Fda Biomarker Qualification Program was added to establish a statutory process for qualifying drug development tools, DDTs, including biomarkers. There are 3 sequential stages of submission and review. The first is the letter of intent. The second is the qualification plan. And the last is the full qualification package. Each stage builds upon the previous stage to support the proposed context of use for that drug development tool. Additionally, each stage consists of a sequential set of reviews by specific staff at FDA, allowing for regulatory engagement throughout the project's qualification process. So you can see that after the letter of intent, there's a 3-month review, qualification plan, 6-month review, and full qualification package is 10-month review. When we look at the letter of intent, this is when the applicant initiates the qualification process by describing the drug development tool, a proposed context of use, and background on the drug development need. The qualification plan, so stage 2 of the 3-stage process, builds upon the letter of intent by addressing recommendations from the FDA expressed in the letter of intent determination letter, but also includes methodologies to support the drug development tool for the proposed context of use. The methods include the data sources, knowledge gaps, proposed data selection plans, and analysis plans. And then finally, the full qualification package is the most comprehensive and final stage in the qualification process. This includes the detailed descriptions of the studies, the analyses, the results, and conclusions to support the context of use for that drug development tool. The qualification process for a project ends with FDA issuing a determination letter for submission with a qualified or not qualified determination. When we look at the EMA qualification process, there are 2 pathways for the applicant. There is qualification advice that is based on the evaluation of scientific rationale and preliminary data submitted. And this advice that's provided from EMA is to help future protocols and methods to refine and develop towards a future qualification opinion. The second pathway is qualification opinion, and this is based on the assessment of the submitted data and conducted analyses. With the 2 pathways described above, these are dependent on the quality and the quantity of the available patient-level data, and this is the primary factor, as well as the analyses that go in to support the context of use for the qualification submission. So I'm going to walk through how the review process works within the EMA qualification framework. The letter of intent and draft briefing document is submitted to EMA from the applicant. This triggers the formal start of the process and the formation of the specific scientific advice working party within EMA. This is also an opportunity to have a preparatory meeting, if the applicant would like, with EMA as a way to optimize the briefing document before the full deep dive regulatory review. Then it goes into the applicant refines the briefing document and resubmits, and now it's the final briefing document that the EMA will use as they start their deep dive regulatory review. After EMA does their review, they can request a face-to-face meeting or a virtual meeting between the applicant and the scientific advice working party as an opportunity to enrich the considerations for qualification recommendations that they will take to the CHMP, which is the Committee for Medicinal Products for Human Use. This also is a time where, depending on the type of data and analyses to support, this may trigger the submission to push towards qualification advice adoption, where SAWP recommends a qualification advice, and then the CHMP will issue that, and then the qualification process is reinitiated. Or it will trigger the draft qualification opinion being issued, in which SAWP recommends a qualification opinion to CHMP, then CHMP issues the draft qualification opinion. Once this is drafted, it goes to public consultation. This is a 6-week period where before final adoption of the qualification opinion for that drug development tool or novel methodology, there's a public consultation by the scientific community. After this, it is the final qualification opinion is adopted by the CHMP. And really the start to finish for the qualification process, from the start to the procedure to adoption of a qualification opinion, is 190 days. What I want to do is walk through the evidentiary criteria and what a table of contents looks like between an FDA full qualification package, which is the final stage of FDA's qualification process, and what goes into the briefing package for EMA for a qualification opinion. You can see that between the FDA full qualification package and the EMA briefing package, the table of contents are very much so aligned and is reflective of the similarities with the background, the context of use, the supporting data, the methodologies, results, conclusions, as well as additional information, including assay information, so understanding the data behind the data. And you can see that they very much so align closely. The other part is that the evidentiary criteria is higher for qualification and even higher for surrogacy. So when we look at the evidentiary standards for qualifying a surrogate endpoint between the FDA and the EMA, you'll see that there's 3 rows. Starting from the bottom is the candidate surrogate endpoint, then it goes to a reasonably likely surrogate endpoint, and a validated surrogate endpoint. When we look at going down bottom to top, the evidentiary standards increase. So when we go for a candidate surrogate endpoint with FDA, this is through a letter of intent, basically showcasing that the biomarker has potential value for an unmet need. When we look at EMA, this can be through a letter of support, say, yes, this is unmet need, encourage further development and validation of the biomarker, and then a qualification opinion may be considered when the results of the final validation steps are available. When we move up to reasonably likely surrogate endpoints, the definition from the FDA is that this is an endpoint that is strongly mechanistically, epidemiologically linked to surrogacy, but without sufficient clinical data to show that it is a validated surrogate endpoint, and that such endpoints may be used for accelerated approval for drugs. So what this means is that with FDA, reasonably likely surrogate endpoint is linked to the accelerated approval program. And you can see that with EMA, there is no such thing as a reasonably likely surrogate endpoint available in the constraints of regulatory considerations for EMA. When we look at the validated surrogate endpoint, this is, by definition from FDA, is providing the strong evidence, clinical data, that an effect on the surrogate endpoint predicts a specific clinical benefit. With EMA, you can see the definition is that there's biological relationship, there's prognostic value of the surrogate for the clinical outcome, and that there is evidence from those clinical trials that treatment effects on the surrogate correspond to effects on the clinical outcome. So this is something to think about as we walk through our qualification submission and tying in the evidentiary standards for both FDA and EMA, and for the context of the data to support the Ibox scoring system. When we look at accelerated approval versus conditional marketing authorization, FDA's accelerated approval pathway is basically linked to surrogacy to demonstrate an effect on the surrogate endpoints that is reasonably likely to predict clinical benefits. When we look at the EMA conditional marketing authorization, the qualifying criteria are that the benefit-risk balance of the medicine is positive. It is likely the applicant will be able to provide comprehensive data post-authorization. The medicine fulfills an unmet medical need, and that the benefits of the medicine immediately availability to patients is greater than the risk inherent in the fact that additional data are still required. So you can see with the accelerated approval pathway with the FDA, this is directly linked with surrogacy, whereas with the conditional marketing authorization, this is not linked to surrogacy. And in fact, conditional marketing authorization is outside the purview of EMA scientific advice working party. So essentially, you can have a conditional marketing authorization without surrogate endpoints through EMA. And this is different than the FDA's accelerated approval pathway that directly links reasonably likely surrogate endpoints with the accelerated approval pathway. So what I want to start to do is transition the information, the learnings covering the regulatory evidentiary criteria for both EMA and FDA, and provide the update on where we are with qualifying the Ibox scoring system as a reasonably likely surrogate endpoint with FDA. This is the current context of use statement presented to FDA. We have an accepted letter of intent in the Fda Biomarker Qualification Program and submitted the qualification plan in April, in which it is under review by the agency. We submitted the briefing package to EMA for a qualification opinion in February and anticipate the CHMP decision in October. When we look at the context of use and this qualification effort, this is the first tool of its kind to seek regulatory qualification for use in kidney transplant clinical trials. The proposed context of use presented to FDA provides a quantitative basis to support the use of the Ibox scoring system at 1 year post-transplant as a reasonably likely surrogate endpoint for the 5-year risk of death-censored allograft loss in kidney transplant recipients for use in clinical trials as an endpoint at a fixed landmark. You'll see at the reasonably likely and accelerated approval refers to FDA terminology. In 2019, the Paris Transplant Group, together with members of the transplant community, published a seminal paper on the Ibox scoring system. The Ibox is a risk prediction tool that utilizes multiple clinically relevant features that have been demonstrated to be mechanistically associated with increased risk of late graft functional decline and failure, including estimated glomerular filtration rate, so eGFR, proteinuria, donor-specific antibody, DSA, and kidney allograft biopsy histopathology to really fully reflect the heterogeneity of graft failure. These measures are taken cross-sectionally at any time point post-transplantation. The Ibox scoring system integrates these measures to generate individualized predictions of outcomes at 3, 5, and 7 years post-kidney transplant. The Ibox scoring system was designed to be used at the patient level to inform clinical care and management of transplant patients. Data was prospectively collected from 4,000 consecutive subjects across 4 health centers in France, and this was used to develop the Ibox scoring system. External validation was performed in cohorts from the US, EU, and 3 independent phase 2 and phase 3 clinical trials. The Ibox scoring system outperforms any of the single components of the Ibox scoring system and each component of the Ibox scoring system has established clinical and analytical validity. The Ibox scoring system in the Lupi et al. 2019 British Medical Journal publication was designed to be used at the patient level to inform clinical care and management of transplant patients. We have adapted the Ibox scoring system to be used as a clinical trial endpoint, incorporating feedback from both FDA and EMA. These optimizations include risk evaluation fixed at 1 year post-transplant. This adaptation was based on feedback from EMA noting that time from transplant is an important prognostic marker, but is never affected by therapy. Therefore, having the risk evaluation fixed at 1 year post-transplant does not impact the output of the model. The follow-up time was set at a maximum of 5 years post-transplant. Subjects who died, lost their graft, withdrew before the first year of transplantation had missing Ibox scores because it was before that 12-month Ibox evaluation. Therefore, analyses were performed, imputing a 1 year Ibox score for these subjects to allow the Ibox scoring system to be used in de novo clinical trials. The imputed Ibox score corresponds to a worst case scenario. So in other words, the worst Ibox score. And this is to prevent survivor bias. And then lastly, in this qualification submission, 2 models are included. One model with biopsy input called the full Ibox scoring system, and the other model without biopsy input called the abbreviated Ibox scoring system. The Ibox scoring system utilizes 4 established clinically relevant patient features that are routinely collected in transplant centers and clinical trials to compute an Ibox score at 1 year post-transplant for the 5-year death-censored graft survival. These features include eGFR and proteinuria in the form of urine protein to creatinine ratio that are measures of kidney function, presence of donor-specific antibody using a qualitative binary mean fluorescence intensity cutoff, and then with or without kidney biopsy histology data using Banff lesion scores. Shown on this slide is the abbreviated Ibox scoring system, so without biopsy. The Ibox scoring system is a composite score with a linear combination of parameters from the multivariable Cox proportional Hodgkin model. The component measures, so for the abbreviated Ibox, eGFR, proteinuria, and DSA were assessed at 12 months post-transplantation and used to compute the Ibox score. Because this is a composite biomarker, obtaining the data sets with the necessary variables to calculate an Ibox score was challenging as expected. For example, urine protein to creatinine ratio was not the proteinuria measurement used consistently across data sets with all the necessary features. Analyses were conducted supporting the conversion from various proteinuria measurements to UPCR for use in the Ibox scoring system models. Another example is the Dsa Mfi. The assay was granted 510(k) clearance by FDA as a qualitative test. Each tissue typing in user laboratory determines a laboratory-specific cutoff for the presence of DSA, and then validates this cutoff according to the package insert for the assay. The optimal MFI cutoff was 1400 based on literature precedence. Thus, we were able to align the analytical intended use of the DSA assay with the clinical utility. We collected the assay information and laboratory certification documentation from the data sets to support this qualification. And then we reviewed the documentation and deemed that the analytical methods were robust, reliable, and fit for purpose. And then lastly, these 5-year death-censored graft survival probabilities were estimated using the 12-month Ibox scores, also known as the survival model. The Ibox scoring system has been developed by estimating individual weights for each of the proposed components at 12 months post-transplant. This table shows the relative contribution of each factor in the model for the abbreviated Ibox scoring system. The predictive performance of the full and abbreviated Ibox scoring system was tested in the qualification derivation dataset. The C-statistic gives the probability a randomly selected patient who experienced an event, so graft loss in this case, had a higher Ibox score at 1 year than a patient who had not experienced graft loss. Both models have high C-statistic values of 0.85 and 0.84 respectively, suggesting they can discriminate accurately within the construct of the proposed context of use. This table summarizes the predictive performance across the validation datasets for the full and abbreviated Ibox scoring system models. Both models have good discriminatory ability across datasets. For the next two slides, I'm going to focus on the abbreviated Ibox scoring system, so without biopsy input, but please note that the predictive performance is similar to the full Ibox scoring system. The predicted versus observed graft losses were not significantly different for the abbreviated Ibox scoring system, indicating good calibration in these data sets. The full and abbreviated Ibox scoring system models have reasonable predictive performance across the qualification data sets to support the proposed context of use. The treatment effect was performed to demonstrate the Ibox scoring system can provide the evidence from clinical trials that a treatment effect on the surrogate corresponds to an effect on the clinical outcome. This was performed on 3 data sets, the 2 RCTs, BENEFIT and BENEFIT-EXT, and a subset of subjects on a specific therapeutic regimen, IMTORI-based. The operating, predictive, and prognostic characteristics of the Ibox score show that the lower the Ibox score values, the better, and is associated with a higher expected death-censored allograft survival. The treatment effect on the Ibox score, which is negative and statistically significant in all 3 trials, corresponds to an effect on the death-censored endpoints, which is also negative in all 3 trials and statistically significant in the BENEFIT RCT. In a prospective clinical trial using the Ibox scoring system, we will need to power to help ensure that the 5-year survival is significant, but these trials were not powered for that. The Ibox scoring system as a composite score is not intuitive. However, the individual components are understood clinical parameters in kidney transplantation and can be used to model a minimal clinically important difference for an Ibox score. So for an example, an eGFR difference of 5 to 10 mils per minute is considered a minimal clinically important difference for renal function. This slide is intended to illustrate how the Ibox changes for difference in Ibox components, and in this example, eGFR. We investigated the expected Ibox score difference if eGFR values were changed and all other parameters remain constant. eGFR is a well-known marker of kidney function, and a difference of 5 to 10 mils per minute is a clinically meaningful difference that has been defined and published. A change of 1 mil per minute in eGFR resulted in a change of 0.046 in the Ibox score, assuming all other parameters remain constant. So a difference of 10 mils per minute in eGFR results in an Ibox score difference of 0.46. And again, a lower Ibox score is better and is associated with allograft survival. So where are we with our analyses, where we are with our qualification submissions, and the immediate next steps? The proposed context of use provides a quantitative basis to support the use of the Ibox scoring system for use as a clinical trial endpoint at a fixed landmark. Qualifying 2 Ibox scoring system models with and without biopsy endpoints will provide sponsors and investigators flexibility in clinical trial design with or without a surveillance biopsy at 1 year post-transplant. Our our team, the C-Path TTC, submitted the qualification plan to Fda Biomarker Qualification Program in April of this year. The qualification plan, so stage 2 of the 3-stage submission, is under review by the agency. And lastly, we submitted the briefing package to EMA for a qualification opinion in February of this year and anticipate CHMP decision in October. When we think about the future impact and this qualification opinion that we anticipate from EMA, it's important to understand the history and the added value of this qualification in kidney transplant clinical trials. Currently, there are only 4 other qualified endpoints with the European Medicines Agency. The Ibox scoring system will be the first qualified endpoint for transplant. Once qualified, the Ibox scoring system will allow sponsors to demonstrate superiority of a novel immunosuppressive therapy compared to standard of care at various time points post-transplant in pivotal or exploratory drug therapy studies. So for phase 2 or proof of concept studies, they can use the Ibox for Ibox assessments at 6 months post-transplant. And for phase 3 studies, this can be where a study duration could be 1 year and up to 2 years post-transplant. In Europe, this would allow labeling claims and promotion of superiority of the new therapy, superiority to the current standard of care, thereby addressing an unmet need in kidney transplant, is a key criteria for conditional marketing authorization in the EU. And this is the first step to enhance development of innovative therapies in kidney transplant. And finally, I want to thank all the TTC members, data contributors, and the C-Path team. We are committed to improving the lives of individuals living with a transplant. Thank you for your time.
Good morning. So hello and good morning. The previous sessions today had been discussing using the Ibox as a reasonably likely surrogate endpoint and kind of some of the stuff involved with that. What I want to talk about now is actually thinking about designing a clinical trial around it. So for the Ibox as a reasonably likely surrogate endpoint, how do we use that to answer trial design questions? How do we figure out what sample size we need to see a difference in Ibox scores? So what we have done is develop a GUI for calculating the sample size that one would need to see a difference in Ibox scores. And this is for a hypothetical trial of 2 arms of a reasonable duration, about 1 year. And there's 2 methods I'll go through. I'm going to go through a standard parametric calculation and an alternative non-parametric method. But first, before I get into that, I just want to say a few things about the parametric method and also what we are not doing, at least in this session. So the parametric method assumes a normal distribution. So it uses that assumption to figure out the sample size you'd need to see a particular difference in mean scores between a treatment and control group. The Ibox is a survival model, so we can actually use it to figure out that 5% 5-year death-censored graft survival in each group, or at least get an estimate of it. But what I want to emphasize here is that the difference in mean Ibox scores that I'm going to be talking about today is what is used to power this 1-year trial, and this is the sample size I'll be talking about today. In another session later, I will discuss the difference in 5-year overall graft survival and what would be needed to power that kind of long-term follow-up study. So just main takeaway here is the parametric method assumes this normal distribution, and I'm going to be focusing on the sample size needed to see the difference in Ibox scores. Okay. So in order to figure out that sample size, we've designed this GUI that should make it easy for a user, or at least that's our hope. So the basics of it are relatively straightforward. A user would start by picking the Ibox scoring system that they're using. So there's 2 versions available. A full version includes biopsy information, and an abbreviated version excludes the biopsy information. We also include 6-month and 2-year time points, but today I'm going to be focusing on that 1-year time point. And then because to start, I'm going to be focusing on the parametric method. I also have that test type equals parametric selected, but the user could also pick a non-parametric test. And then the user would figure out that Ibox score difference by calculating it directly from the control and treatment groups based on their parameter values, which I'll show on the next slide. The variance gets calculated directly from the data, and that'll be shown in 2 slides. And then there are some standard inputs to pretty much any sample size calculator, things like a superiority margin, type 1 and type 2 error rates, and the allocation ratio. And these can be adjusted by the user as needed. Okay, so calculating the Ibox score difference is done via this calculate button. And we realize that the actual clinical meaningfulness of an Ibox score difference can be a little bit opaque. So it may not be obvious what negative 0.48 means for an Ibox score difference. But so the way we have this set up is that the different parameters in the Ibox, which have a more clear clinical meaning. So these are, for the abbreviated Ibox, estimated glomerular filtration rate, which is EGFR, the urine protein creatinine ratio, which is UPCR, and the donor-specific antibodies, which is DSA. These have clear clinical meanings, and we allow the user to adjust these, and then these are taken and automatically generate an Ibox score difference. So the example I have here shows a difference in 10 EGFR between the treatment and control groups, which is thought to be a clinically meaningful difference in EGFR, and UPCR and DSA are held constant. So those are the same in each group, and this is just a difference in 10 EGFR, and that relates to that negative 0.48. And the reason it's negative is that lower Ibox scores indicate lower risk of graft loss, so the lower scores are better. And then we also have the full version that's available, which looks just like this, but it would also include the biopsy inputs. So things like IFTA, transplant glomerulopathy, and a few others. And then once the user has set the Ibox difference, they can adjust the variance. And this is done mostly without user input. The variance is calculated directly from the data, but the user can select different patient characteristics to exclude. So a user could exclude patients that have donor age greater than or equal to 60 years. They can exclude previous kidney transplant, etc. And what that will do is it will automatically update the variance in blue there at the bottom right. There are also some patients who we impute values for. So when we're measuring an Ibox score at 1 year, that measurement comes from the 1 year data. If a patient in a trial has a graft loss, a death, or a loss to follow-up, and they don't make it to that 1 year time point, we can't get an Ibox score for them at 1 year. So what we do is we impute a value that indicates very poor performance for them. For the abbreviated Ibox here, that's 1.48, and the user can set the percent graft loss, death, and loss to follow-up. So this is a total of 5% imputed values, and that also affects the variance. And as those change, the variance will change with them. Generally speaking, the more imputed values you have, the higher the variance is going to be. And then once all those inputs are set, the user can click run, and this table will get populated. This table functions in a way where a user can actually remove rows if they want. So the red button, a user would click on a row, it would highlight, and then the user can click the red button and that will delete the rows. Or if the user wants to export the table, that can be done using that yellow-orange button. The main output to focus on here are the mean Ibox difference. So this is kind of the main thing that's driving the sample size, and the sample size estimates. And then the other inputs are still stored on the right. So you can see variance, superiority margin, the type 1, type 2 error rate, allocation ratio. And these estimated sample size will change with the different inputs. So we can see here that for one of these rows, I selected this Ibox scoring system that to be full, whereas the others are abbreviated. We can also see that the first 2 rows have a type 2 error rate of 0.2, which corresponds to 80% power. And the first row has a 2-to-1 allocation ratio. And these cause some fluctuations in that estimated sample size. And one thing to just re-emphasize here, I mentioned this earlier, but I just want to make sure it's clear, is that this sample size is for that 1 year mean Ibox difference. The GUI also provides an estimate of the 5-year survival difference for the allografts, but this is not the sample size that's given. To see a difference here, a larger sample size would be needed, and I'll be discussing that more in a later session. Okay. So now I'd like to also give a demonstration of the GUI. So just a live demonstration here. So let me just bring this up. If you see me looking way off, that's just because I have multiple monitors here. So let's bring this up. Here we go. Let me refresh. Okay, so what I want to do here is just give a kind of live walkthrough of how to use this GUI. So the way this works is when it first pops up, the sample size calculator gives the option to take a tour. And this tour kind of just takes a user through the different options here. So you can select the different parameters. It shows you, like, you can pick the different Ibox version here, takes you to the calculate button, reminds you, says what it is, adjust variance, run, shows the output, and the different buttons that can be used. And now, of course, I went through that very quickly because that's something that a user could do on their own time. I just want to give a quick demo here. I also want to note that in case anything here is confusing, there is an about page that helps explain things and a user guide that will hopefully be useful to a user who has any questions. Okay, but I just want to run through a couple of examples here. So we can choose between a full and abbreviated Ibox, and what that's going to do is it will change the variance, which, because the data is the variance is calculated directly from the data. So it's calculated directly from data where we have run Ibox scores on for the full and abbreviated versions. But it will also change the inputs here. So for the full Ibox, it includes all of these biopsy variables. So there's just a lot more to input with the full Ibox. So to keep things simple, I'm going to stick with the abbreviated Ibox. There are also other time points. These will affect the variance calculation. But I'm going to focus today on this 1 year time point. And for now, I'm going to stick with the parametric test. So a user can input these values here, change the EGFR, UPCR, and DSA. For today's demonstration, like in the slides, I'm going to stick with this 10 EGFR difference and keep everything else the same. And that translates to this difference of negative 0.48 in Ibox scores. We can also adjust the variance. So let's say we want to, let's say we want to exclude donors that are greater than or equal to 60 years old. You can see that this variance updates automatically. So if we include those patients with the donor age greater than or equal to 60, the variance is 2.21. When we exclude them, it automatically updates. And let's say we also want to exclude previous kidney transplant and DSA at time of transplant. That gives us an adjusted variance of 2.2. Also, we have these imputed values. I mentioned these are needed to account for the patients who don't make it to 1 year, and that the more imputed values we have, the higher the variance. So generally speaking, for a 1 year trial, we shouldn't have too many imputed values, but maybe we assume that these defaults are a little bit high. Maybe we think that the deaths aren't going to be quite this high. Let's lower the deaths. What we can see is that this variance automatically adjusts and goes down. So fewer imputed values leads to lower adjusted variance, generally speaking. So let's set that. We've got a superiority margin of 0. I think that's fine for now. Type 1 error rate of 0.05. And let's make this type 2 error 0.2 instead. We've got a 1-to-1 allocation ratio, and we can hit run, and then this table automatically populates. And we can see the estimated sample size. And there's other things we can do, like maybe we want to switch this type 1 error rate to 0.1 and hit run. That will affect things. We could add in a superiority margin. So this is kind of a minimal difference you want to test against, like a minimal clinically relevant effect. Maybe we just want something relatively small. Let's say it's negative 0.1. That affects the sample size. And yeah, that's basically it for the live tour. Users can also, let's say these other ones we ran, maybe we don't want to keep these. So user can select these and delete as needed and export anything that they want to save. Okay, so that concludes the live example of the parametric version. Let's go back to the slides and I'm going to get into the non-parametric version now. So the parametric approach, and as I was mentioning, the imputed values, the more of them you have, the higher the variance gets. So a parametric approach assumes a normal distribution. So it's using a difference in Ibox scores, assuming this normal distribution, with a known variance and these percent imputed values to figure out the sample size. And these imputed values are necessary. We have to have them because we have to account for patients who don't make it to 1 year, because we don't want our results to be affected by survivor bias. So when we include these imputed values, the more of them we have, the higher the sample size gets because the variance gets bigger. So what's basically happening is these imputed values indicate very poor performance because these patients didn't even make it to that 1-year time point, and that effectively introduces outliers into our data set. So what's happening is that we no longer really have a normal distribution anymore. We have these extra values that are getting added in that are very far from kind of the means of the distributions, and that is raising the sample size and making it so that the parametric test is no longer the most efficient test. When we have imputed values, a more efficient test is actually a non-parametric test, which doesn't make any assumptions about the normal distribution, and it relies on ranks rather than kind of the distance from the means. So when you impute a value and you add in this kind of outlier, a non-parametric approach is less sensitive to that because it's just the lowest rank. The actual distance isn't what matters. What matters is what rank is it. And a non-parametric approach is still a very effective test of difference between two groups. It's still a very powerful test. So the example I have here on the bottom, I'm going to go through the table from left to right. We have our 2 randomized control trials, the benefit and the benefit extend studies. The treatment group for the low-intensity belatacept arm for the mean Ibox score is given there in the next column, along with the control group, the cyclosporin arm, their mean Ibox scores. We can then see the difference in Ibox scores between groups along with the variance. And then we can see a significance test. So this first one here, the second to last column, is the 2-sample t-test, which is a parametric test. It assumes a normal distribution. And we can see for each group, we're finding a significant difference between the groups, assuming that p-value of 0.05. With the non-parametric approach, we also see the same significant difference. This is a Wilcox and Whitney test, and it doesn't rely on those normal distribution assumptions. It is just looking for ranks and seeing whether the ranks between the two groups are different. And it is saying, yes, they are. So the type of test one chooses at the end, if one wants to use a non-parametric test, it's still an effective test. And in the presence of imputed values, it might even be more efficient than the parametric test. So what I'm going to do here is I'm going to go through a little bit of how we actually do this non-parametric approach in the background. From the user perspective, it's going to be very similar to using the parametric approach, but I want to explain a little bit of what's going on in the background. And then at the end, I'm going to compare the 2 approaches to see which gets the lower estimate of sample size, given certain inputs. So for a non-parametric test, we need some distribution of ranks. For a parametric test, we don't need a full distribution. We can assume it and just calculate based off of a difference in Ibox scores between the two groups. But for the non-parametric test, we actually need that distribution of ranks. So we're doing something a little bit different where we're asking, is the probability that a rank in the treatment different than the rank in the control? So the way we approach this is we simulate distributions. I've got an example of Ibox score distribution on the right from our data. We can see that for everything that if you exclude the imputed values, they look relatively normal. But when we include the imputed values, we've got this extra mode. So we can use that information to simulate distributions. And once we've simulated a large number of patients in each distribution, we can use the non-parametric test to figure out the kind of sample size we need to see some similar distributions of ranks. So from a user's perspective, everything is basically the same. But what's going on in the background is a little bit different. So for a parametric test, the user inputs here are used to figure out that Ibox score difference. For a non-parametric test, it's slightly different. Now these user inputs are used to figure out the treatment and control distribution means. The variance is then taken, not counting the imputed values, to get the variance for each distribution. And from that information, since we have this treatment distribution mean, control distribution mean, and variance, and we know that from our data that these are approximately normal-ish distributions, we can simulate large number of patients from each group. And then from the user inputs based on those imputed values, we can add on the percent imputed values to each group. And that gives us a full simulated distribution. And from that information, we can estimate sample size. So now I think the main question is, what is the actual difference between these two approaches? If we have a 1-year trial where we've got this 5% imputed values, what gives us the better estimate of sample size? And I've made some assumptions here. We've got this 2-sided alpha of 5%. We've got 80% power. We've got a 1-to-1 allocation ratio. And as I said in the question, we are including those imputed values for early events. And for each group, the only difference is this difference in 10 EGFR, which translates for the full and abbreviated Ibox to a difference of negative 0.46 for the full and a negative 0.48 for the abbreviated. We also have the pooled variance from the two groups. And then this second column from the right, this 4 is the parametric total sample size estimate. And then the final column on the right is the non-parametric total estimate. And what we can see here is that the parametric and the non-parametric estimates are quite different, and the non-parametric estimate is a lot more efficient. So if a user wanted to run a trial and had to deal with these imputed values, and the number of patients one would need to find a significant difference between groups using a non-parametric test is quite a bit lower than the patients one would need to see a significant difference using a parametric test. So the non-parametric test is actually quite a bit more efficient in the presence of imputed values. So I'm just going to do another quick demo here, just showing that's the non-parametric method. So let me bring back up the GUI. Here we go. So I'm going to keep everything exactly the same as before. So this was the output we got. Actually, I'm just going to rerun it so we can see that it is the same. So this is the same output as before. And now I'm just going to switch the test to non-parametric, and I'm going to hit run again. And what's going to happen now is we'll just see here, given the exact same user inputs in the GUI, the only thing that's changed is the non-parametric test. We can see how the GUI actually works. And the main thing I'm showing here is just, we can see that there's this calculating thing showing up. This is just because we're doing a simulation, and it takes a little while to run. It takes about 30 seconds to a minute. But before too long, it should finish, and we will see the sample size estimates. And as we'd expect before, again, the user inputs here was, I think, 4% imputed values. So we should expect the non-parametric estimate to be a little lower than the parametric estimate. Okay, so let's just let this finish up. It should only take a moment. There we go. And as we can see, the non-parametric estimate is 232 versus the parametric, which is 360. So in the presence of imputed values, that non-parametric test is just a bit more efficient. Okay. So finishing up here, just a few conclusions. We've developed a sample size calculator to assist with clinical trial design using this Ibox as a reasonably likely surrogate endpoint. It's available for both the full and abbreviated Ibox scoring systems. And again, the difference between those is that the full is the biopsy information, and abbreviated is without biopsy. It incorporates parameters that are easily adjusted to help make the user, make things easy for the user in terms of making informed decisions. It is driven by the mean difference in Ibox scores to evaluate drug efficacy between a treatment and control group. We can also account for those patients who don't make it to that first year because they've died, lost a graft, or lost a follow-up using imputed values. And then we implement both the parametric and non-parametric approaches to help maximize efficiency, because in the presence of imputed values, the non-parametric approach tends to be more efficient. But if there's very, very few imputed values or close to zero, the parametric approach is probably going to be a little bit better. But with those imputed values, the non-parametric approach is a little more efficient. And that's about it. We hope this was informative for how to use the GUI, and we hope you enjoy the rest of the sessions today.
All right.
Thank you very much, Amanda and Luke, for your excellent presentations on the Ibox as well as the GUI. We have several questions, and please continue to enter your questions into the question and answer box, and I'll review them and pose them to both Amanda and Luke. So first, the first question that I want to pose is to Amanda. And the spirit of the question is, if we had the Ibox available either in the past or as it becomes available now in the future for clinical trials of new transplant immunosuppressants, how do you think it might change the uptake of new immunosuppressants after they're launched?
Thanks for the question. So the benefit of this reasonably likely surrogate endpoint is that you can demonstrate superiority of the novel immunosuppressive therapy at 1 year, with the stipulation that you, through accelerated approval, that you have the continued monitoring to show the outcome at the end of the accelerated approval pathway. So you'll be able to demonstrate superiority earlier on at 1 year, but requiring the continued monitoring through the accelerated approval pathway.
Great, thank you. And Luke, a couple of questions on the sample size methodology and the GUI. First, can you tell us how it will be available to people who want to use it, and have you published the power and sample size methodology to the Ibox?
So it should be available as just a web application. So someone could, it's through rShiny, so that's just all on the website. So as long as you have the link to it, you should be able to access it. And as for publishing it, we're actually working on, we haven't written the paper yet, but we are looking to kind of get a paper going and publish kind of a walkthrough of it. So we're working on that.
And just to piggyback that, I was going to say that part of this contract and the intent of the TTC is to collaborate to make things publicly available. So once qualified, the Ibox scoring system will be publicly available, and the graphical user interface for the sample size calculator will also be publicly available. And so post this workshop, we will be writing a publication, but separate from that, as Luke mentioned, we are also drafting a publication on specifically the sample size estimates.
Great. Thanks, Amanda and Luke. Also for Luke, you mentioned the difference between choosing a parametric versus a non-parametric test, and you were using the 5% graft failure or death in the first year. Is there a crossover point, 2%, 3%, 4%, at which a parametric test might be more appropriate than the non-parametric?
Yes, there is. I've actually plotted this out using an example with certain parameters. I need to remember exactly what those parameters were, but when I was looking at this, when plotting it, the crossover point was at about 2.5%. So if you have more than that, so if you have like probably more than 3%, it starts getting more and more efficient to just use that non-parametric version.
I see. And is it substantial in that if you only were having 0 to 2% in your trial, anticipating your trial, you would recommend using the parametric over the non-parametric? Yeah,
I say 0 to 2%, 2.5%, you probably might as well just stick with that parametric. Once you start getting much over that 3%, at least from the example I've looked at. So it might be that with different examples, it might change the crossover point. But from the example I looked at about 3%, it starts getting notably more efficient to just use the non-parametric version.
So in terms of application, would you say that means as you look at, as a sponsor or investigator looks at planning their kidney transplant trial, depending upon the risk that they see in the population that will be included, if it's a very low risk group with an anticipated small, low % of deaths or graft losses in that first year, they may opt for the parametric, whereas if it's more standard risk or higher risk group, the non-parametric would be more efficient. Is that a logical conclusion, Luke?
It sounds logical to me. The only caveat I'd add is that I don't know exactly what the percentages are for the kind of standard or higher risk, but assuming that you think it's going to be, much more than 3%. So if you think it's going to be 4%, 5%, maybe more than that, then the non-parametric should give you a pretty clear benefit.
Great, thank you. Now a question for Amanda. Amanda, will the FDA, under the reasonably likely surrogate endpoint, allow for superiority claims of Ibox in labeling information, or is that just the EMA? So if it's qualified as a reasonably likely in the U.S., will we have superiority claims in the U.S. label?
So, it's under the assumption that it is qualified, and that will be That will have to be part, if it's part of the primary endpoint. That's the potential, right? That's the vision as of reasonably likely. So yes, in a label, sponsors should be able to describe the benefit that they observed with their novel therapy, showing superiority.
Okay, great. And from my perspective, that's one of the great advantages. If it's a reasonably likely surrogate qualified primary endpoint in a FDA approval, it will be in the label. It'll be the basis of the approval and therefore can be promoted by the sponsor at the time of approval.
Right. And with the EMA, it's different, right? And this goes back to the dialogue of how we've really leveraged our understanding of the regulatory, the clinical, the statistical between EMA and US to have as much alignment as possible, but with qualification as a secondary endpoint, knowing the stipulations with a fully validated surrogate, sponsors can still promote superiority in their label claims as a secondary endpoint in EMA with Europe, in Europe.
Excellent. Thank you. And then I think this will be our last question for Luke. And it regards the GUI again and calculating sample size. If you're, for instance, doing a study with no preformed DSA and it's TAC-based immunosuppression, the control arm, then the tool can only use these as reference for the sample size calculation. In other words, with the exclusion tool, can you restrict the data set used to calculate the sample size to the type of population you want to test?
As long as there's we have that filter available, I need to double check as to which. I think we have all those filters, but yeah, as long as you have those filters available, you should be able to exclude those populations, and then the variance will be calculated from our data without those patients. So yes, that's exactly how it would work. And there was also one quick thing I thought of for a previous question about the when is the kind of the parametric and non-parametric more efficient. So I just want to add a quick thing to that, which is that what I said is kind of assuming you've got this nice normal distribution. In theory, if you, for whatever reason, and we haven't really seen this when we've looked at data, but if for some reason you had data which was very much not normal, even if you didn't have a large number of parametric, if you didn't even have a large number of imputed values, you might still find that the non-parametric could be more efficient. But so I just wanted to add that caveat on that. This is, given a hypothetical situation based on some of the stuff we've observed.
Thank you, Luke. And thanks, everyone, for entering questions. Please continue to do that during the upcoming presentations, because this is what we'll use for the panel discussions. So I appreciate that. With that, let's move on to Session 2. Session 2 will focus on the use of real-world evidence in transplantation trials. Thank you.
Hello, everyone. So in a previous session, I discussed the sample size needed to detect a difference in Ibox scores. In this session, I'm going to discuss the sample size needed to detect a difference in survival for a long-term follow-up study. So just to start with a really brief outline, I want to just cover kind of the really basic methodology of estimating sample size based on survival. And this is going to be a very brief overview. I'm not going to go into the details, but I just want to give enough of an idea to kind of understand why a estimate of a difference in survival can get large. The sample size required to see that difference, why that might get large. And then I will cover the 5-year sample size requirements of survival for kidney transplant recipients. So if you recall from my previous session, I had discussed the GUI, and the GUI is designed to calculate sample size for a 1-year trial designed around Ibox scores. And that is shown in that box in the middle. So for this Ibox score difference of negative 0.48, we get a particular estimate of sample size that would be required to see that difference given the rest of the inputs like variance, type 1, type 2 error rate, etc. But what we also want to know, because this is for a for a reasonably likely surrogate endpoint as part of a accelerated approval process, we'd also need to be able to demonstrate superiority in 5-year graft survival, because that's the true endpoint. So we want this biomarker that indicates the composite biomarker, that's the Ibox, that indicates that there's going to be an improvement in survival at 5 years. We need to actually demonstrate that that 5-year survival improvement is there. One thing I want to note additionally here is that this estimate that's shown in the GUI is for death-censored allograft survival. However, what we actually need for that 5-year endpoint, this is what's accepted by the regulators, is overall graft survival, which includes death as an event. So this estimate here is going to be a little different than the estimates I talk about later in terms of figuring out that difference in survival between a treatment and control group for kidney transplant patients, say after 5 or so years. Okay. So the very, very basics of what are required to figure out sample size for a difference in survival need a couple things. So we need to know the number of events that are required by that difference in survival. So the difference in survival gives us an estimate of the number of events. And that estimate also depends on the type 1 error rate, the power, the allocation ratio. And then we also need the probability of seeing an event. And if we can figure out those things, the calculation itself is relatively straightforward. It is just the number of events we need to see divided by the probability of seeing an event. However, actually calculating the number of events and figuring out the probability of event can get a bit tricky. So I'm not going to go into the details, but I do want to at least give a sense that the required sample size is a function of these two things. If you have a large number of events and the probability of seeing an event is small, the sample size requirement can get very large very quickly. Or if you just have a very high number of events that you need to see, that can also result in a large sample size requirement, even if the probability of seeing an event is relatively high. Okay, so I want to kind of revisit a scenario that I showed on that earlier slide where we've got this difference in Ibox scores of negative 0.48. That corresponds to that 10 milliliters per min EGFR difference between groups. We have this 1-to-1 allocation ratio, 80% power, and a two-sided alpha of 5%. And on the right, I've got the sample size estimate. So for the parametric and non-parametric tests, you can see that it's 300, 200-ish. And then as I go, I'm going to try to fill out the bottom row, which is the 5-year overall survival difference. Okay. So the first thing we need to know is, what is the treatment improvement over control? From past studies, from phase 3 kidney transplant clinical trials, we can get maybe a little bit optimistic estimate where we can say that this should correspond to about a 90% treatment and 85% control survival. And keep in mind, this is for overall survival, so it includes death and loss to follow-up. And as I said, this is maybe a little bit optimistic. It might be like 4 to 5%, so we're a little bit on the high end. But this is based off of what we know from past phase 3 kidney transplant trials. And then from that information, we can figure out both the number of events, and I'm going to give a very crude estimate of the probability of events. So the number of events we need to see, and again, I won't go into the calculation for this, but it is about 167. And then the probability of the event, this is a kind of gross simplification, because we could actually, in terms of figuring out the probability of the event, we could look into things like, what is the dropout rates, and there's other things we consider, like the accrual period of a trial. But assuming we know the event rates at 5 years, that's 10% in the treatment, 15% in control, we can get a really kind of simple estimate of that probability. However, I really want to stress here that this probability of the event that I've done here is very likely a optimistic estimate. This is probably, in reality, given the fact that we have dropout rates and other things, this is probably an overestimate. So what it's going to turn into is a very optimistic estimate of sample size. So the actual calculation here, once we figure out the number of events and probability of event, is pretty straightforward. It's just the number of events divided by the probability of the event, and that gives us a total sample size of 1,338. And again, this is an optimistic estimate. This is a low estimate of sample size, probably lower than what would really be needed. But I want to just give an example of that optimistic estimate, because even with that optimistic estimate, if we're looking at the other types of tests, so that the sample size required for that 1-year Ibox score difference, that survival sample size is more than double, more than triple, it's more than quadruple that. It's quite a lot larger. And what this basically means is that we need some way to get additional patients to supplement probably just the control arm for clinical benefit verification when using the Ibox as a reasonably likely surrogate endpoint as part of the FDA's accelerated approval program. So basic take home here, that sample size for the 5-year survival is going to be quite large, and we need some way to supplement patients. One way might be to recruit additional patients, but unfortunately, that's at least somewhat impractical. Running a 5-year-long study with 1,300 or considerably more patients. So some of the inputs I put into this when I was making these slides, I got back sometimes like 3,000 or 5,000 patients for inputs that aren't that different than the ones I showed. The ones I showed are a little bit optimistic. So designing a 5-year-long trial with over 1,300 patients to demonstrate the improvement of graft survival is probably not very practical. So there is a solution, and that solution is to draw from real-world evidence. So registry data such as UNOS can be used to supplement the control arm and attain the required sample size. However, there are regulatory and design considerations that we need to be aware of when we use real-world evidence to supplement a transplant clinical trial. And those considerations are important, especially for the regulators, to show that the supplementation is like the patients drawn in from the real world evidence are comparable to those of the current study. However, for this talk at least, I'm actually not going to go into that. This is something that's going to be discussed in the other talks during this session. So I hope you stick around for those talks, and I hope you find them interesting as to the use of real world evidence in clinical trials. And I hope what I've managed to do here today is just show that using real world evidence for such a trial, for a transplant trial, for part of accelerated approval, that we probably do need real world evidence patients for that long-term 5-year follow-up to demonstrate that true difference in graft survival.
Hello, thank you for attending this session. Today I'll be discussing how we're leveraging registry data for an externally controlled trial in transplantation. For those of you less familiar with transplantation, OPTN stands for the Organ Procurement and Transplantation Network. In addition to managing the country's waitlist and matching organs to potential recipients, we also maintain all the data in a registry. I work for UNOS and we serve as the government contractor managing the network. So why would we want to use a historical control? Long-term outcomes require large sample sizes, much larger than you're likely to recruit in a clinical trial, never mind retain for 5 or 10 years down the line. By leveraging real-world evidence, we have hundreds to thousands of potential controls available to us. Additionally, registry data allows us to investigate multiple comparison groups. It allows us to perform sensitivity analyses by looking at different treatment regimens without needing to recruit more patients. In particular, since we're interested in long-term outcomes, using registry data allows us to update the control arm as consensus around standard of care changes. The benefit study was a randomized control trial looking at immunosuppression regimens in kidney recipients. It consisted of 3 arms: belatacept moderate intensity, belatacept low intensity, and cyclosporin, which served as a control arm. Each arm consisted of about 225 to 230 patients. For our real-world evidence study, we are using registry data to serve in place of the cyclosporin control arm. The trial spanned both the US and Europe, so we expect the populations to be different since our registry data is limited to the United States. In a few slides, we'll discuss how we handled that. In order to select our control arms, we identified patients from the OPTN registry who met the study inclusion and exclusion criteria as defined on this slide. The cohort is limited to 18 months, a relatively short period of time. It's open to both living and deceased donor transplants, but certain donor conditions could exclude the transplant from the study. Recipients could also be excluded for high sensitization, defined using PRA panel reactive antibodies. As I noted earlier, the advantage of using registry data is that we have multiple comparison groups we can work with. The benefit study looked at low and moderate intensity belatacept. You could look at either of those or both of them combined. Today's discussion focuses on belatacept low intensity. For our control groups, we will look at cyclosporin, which was the control used in the trial. We will also look at tacrolimus, an alternative immunosuppression regimen, which is also accepted as standard of care treatment. We can further define our comparison groups by induction regimens. The benefit study had patients use Simulect induction. To broaden our sample size a bit more, we created cohorts where either Simulect or Zenipax induction were used. This slide describes the 4 cohorts from the registry data we will focus on today and their respective sample sizes. Because cyclosporin was used as the trial control arm, we had to exclude any patients from trial centers to ensure there was no overlap. For tacrolimus, we could include any patient, regardless of what center they were transplanted at, because tacrolimus was not used in the original study. Because this isn't a randomized trial, we can't assume that our treatments and controls will be balanced as if they were randomly assigned a protocol. Instead, we look to propensity matching. With propensity matching, we find treatment and control pairs that are similar to each other in terms of the characteristics on the right. After matching, overall, our groups should be balanced. This helps us to control for confounding variables so we can focus on the treatment effect. We used R, a statistical software, to perform the propensity matching, using the specifications described on the slide. Today, I'll be discussing the results from the matching, but we will add propensity weighting analyses in the future. I'm going to walk through a little bit of how propensity matching works in our study. Here's an example using the OPTN tacrolimus control. On the left, we have propensity score logits for the OPTN and benefit transplant. The common support area is represented in the shaded blue box. Our matching will be limited to this region. Then, individually, the cases and controls will be matched one-to-one with a 0.2 caliper width, which is a measure of how similar or different we'll allow the matched pairs to be. On the right, we have the results of the matching. The blue box is who was included. Almost all of the benefit transplants labeled as treated matched. There were hundreds more OPTN control transplants, so many of those did not match. This table describes the sample sizes after matching. When working with cyclosporin controls, there were less suitable matches, so our sample size is reduced. Because we had over 1,000 tacrolimus controls to choose from, we were able to match almost all of our belatacept treatment recipients to a control. Ideally, after propensity matching, our samples would be balanced, so we'd only have to focus on the treatment effect. Unfortunately, with these small sample sizes, there were some variables we have to be mindful of. The red line shows how balanced each of the potential confounders are before matching, and the teal line shows after matching. Ideally, the teal line would be constrained by the vertical dashed lines. The dashed lines represent a 0.2 standard mean differences, which is the maximum acceptable difference between treatments and controls. If a difference falls outside of the dashed lines, we need to adjust our regression models. So using OPTN cyclosporin with Simulect induction, which is the top left panel, we're going to have to adjust for recipient age and HLA mismatch because those had differences that were outside of the 0.2 width. The first outcome we'll look at is graph survival. Here we have Kaplan-Meier curves. The red lines are the matched belatacept recipients, our treated group. The blue lines are the OPTN control, which are defined in the blue box at the top of each plot. The top two plots are cyclosporin, and the bottom two are tacrolimus. On the left, we have Simulect induction, and on the right is Simulect or Xenopax induction. For each of the plots, the green line does not change. That is the cyclosporin control arm from the study and is provided on the plot for reference. The belatacept line only changes slightly due to who was matched in each cohort with the OPTN external controls. We compare the survival curves of the belatacept and OPTN control arms using a log rank test. We do not have any statistically significant differences here, but we can still discuss the trends that we see. In general, we see the OPTN control arm is very similar to the cyclosporin arm from the trial. On the bottom right, we see a bit of separation between these two groups, indicating there may be a survival benefit of tacrolimus with Xenopax over cyclosporin with Simulect. However, the heart of the story is that the real-world evidence external controls are generally performing similar to the control arm from the trial. This provides another way to look at the survival. Here we have point estimates at 5 years. Again, the green line on the left, or the green point on the left, is the cyclosporin control from the study, and the blue is the OPTN controls. Belatacept is in red. The next analysis we looked at was death-centered graph survival. Our standard analysis protocol is to consider any death also a graft failure. However, this analysis censors patients who died with a functioning graft. With this approach, there are less events, which makes it look like the red line for belatacept is missing from the top left panel, but I promise you it's there. All the graft failures were just early on, and the line is hidden under the others. Belatacept had very high survival in all cohorts, but was only statistically significant compared to OPTN cyclosporin with Simulect or Xenopax induction with a log rank test. That's the plot on the top right. The benefit cyclosporin controls generally fall in between the belatacept and OPTN arms. And again, this is another way to look at the same data. We're looking at the point estimates at 5 years. And finally, we have patient survival. Generally, patient survival is very high for kidney recipients, so it's understandable that we do not observe significant differences here. However, all cohorts showed the same trend, with belatacept having the highest survival. And just one last view of this with the point estimates instead of the curves at 5 years. The Kaplan-Meier models we were discussing in the past few slides are non-parametric models. The Cox model is a semi-parametric model and describes the risk or hazard of an event in one arm relative to the other. Here the hazard ratios are for the belatacept group relative to each of the controls. Belatacept relative to cyclosporin from the randomized control trial is on the left, and the other estimates are for belatacept relative to the real-world evidence historical controls in the middle and right panels. Recall that in a randomized control trial, we can assume samples are balanced, so we do not need to adjust for potential confounders. We tried to create balanced samples with our historical controls by using propensity matching, but for each of the models, there were still a few unbalanced variables. Those have been adjusted for in each of these models. Graft survival is shown in the top row here. The results are very similar to what we saw in the Kaplan-Meier models, where the real-world evidence estimates are very similar to the clinical trial, except for tacrolimus with Simulect or Xenopax induction. The hazard ratio estimates are below 1, indicating a survival benefit to belatacept recipients over each of the respective controls. For death-censored and patient survival, the results are more mixed in terms of strength, but all indicate a survival benefit for belatacept recipients. For a different outcome, we have delayed graft function, which is when dialysis is needed within the first week after transplant. These results from the logistic regression model are presented as odds ratios, where odds ratios less than 1 indicate improved outcomes. The rates for delayed graft function were about 13% in our cyclosporine controls and 10% in our tacrolimus controls. And the benefit trial data, cyclosporine controls had a delayed graft function rate of 17.6%, and belatacept low intensity was 13.7%. These results suggest delayed graft function rates are lower for belatacept in the randomized controlled trial data and higher when compared to the real-world evidence historical controls, but neither are statistically significant. This aligns with what we saw in the trial results. So moving forward, we're going to continue looking at the propensity matched analyses and round that out with eGFR, estimated glomerular filtration rate. And then we're also going to repeat all of those analyses, the Kaplan-Meier, the Cox models for survival, the eGFR, and the delayed graft function in a propensity weighted framework. So in summary, what have we learned about using real-world evidence to form a historical control arm? First off, you have hundreds of thousands of controls to choose from. You can even include multiple control arms if you want to perform sensitivity analyses. On paper, the belatacept recipients and OPTN controls were very different, but we were able to extract a subset population that was more similar using propensity matching. Moving forward, we're going to expand on this with propensity weighting. However, even after matching, the samples were a bit unbalanced. This is probably due to the benefit studies spanning both the US and Europe, which are very different transplant populations. However, we can still adjust for the unbalanced potential confounders in our model. In doing all of this, we were able to produce similar results as the benefits study trial when looking at the survival rates and delayed graft function. We've exemplified that using real-world evidence does work, and we will look forward to seeing how it can be used in future trials. Thank you for attending this presentation. If you'd like to reach out to me with any questions, my email address is available on the screen.
Thank you very much. I'm John Concado from the FDA, and I'll be talking about regulatory and design considerations for real-world evidence. The views and opinions expressed are mine and should not be attributed officially to the FDA. I do not have any conflicts of interest to report, and mention of commercial products should not be construed as an endorsement. As an outline of what I'll be discussing, first I'll provide an overview of FDA's real-world evidence program, focusing on guidance documents and development projects. I'll briefly describe a conceptual approach to real-world data and evidence. I'll next discuss FDA's experience with real-world evidence submissions for drugs and biologic products. And I'll wrap up with identifying challenges and potential contributions of using real-world data and real-world evidence. As a starting point, we're here mainly on the basis of 21st Century Cures Act of 2016, where FDA was asked to and did develop a program to evaluate the potential use of real-world evidence to support a new indication for a drug already approved or to satisfy post-approval study requirements. As virtually everyone in this audience know, we published a draft framework in 2018, which we're still following, and we did publish draft guidance late in 2021. Just to mention, our standard for substantial evidence, as you'll hear again later, remains unchanged, and we have met our commitments under the Prescription Drug User Fee Act 6 as we ramp up for PDUFA 7. Again, hardly needs a reminder, but these are the FDA definitions from our 2018 framework on the left. Real-world data are data related to patient health status or delivery of healthcare, routinely collected from a variety of sources. We often immediately think of EHR data, claims data, registries, etc. On the right, real-world evidence is clinical evidence regarding the usage and benefits risk of a product derived from the analysis of RWD. So a very straightforward definition. The point I'll emphasize here that sometimes folks forget is that randomized trials can generate real-world evidence when they incorporate real-world data. We often think first of externally controlled trials, if not first and foremost observational studies. Wrapping up with this overview, what I'll be talking about applies to the Center for Drugs and the Center for Biologics, and by extension, the Oncology Center of Excellence. The Center for Devices and actually other centers at FDA have their own robust real-world evidence programs, but obviously there are some differences in regulations and therefore differences in approaches. I'll spend the next few slides just providing a little window into the internal processes, external stakeholder engagement, guidance development, and demonstration projects to place our discussion in context. First, combining the internal and external engagement, I want to point out the real-world evidence subcommittee has a lot of internal activities that aren't visible to the outside world, but it's comprised of membership of FDA staff from multiple CDER and CBER offices. And the committee serves to provide oversight of policy development, including guidances that I'll be mentioning, as well as offering resources and leadership, say, to review divisions. In terms of external activities, we provide feedback on novel approaches that are brought to our attention by sponsors, data vendors, et cetera. And we also discuss initiatives that are presented to the subcommittee for consideration. Additional activities, and this is only a partial list, would be holding FDA or center-level public meetings on relevant topics and conducting FDA small business and industry webinars or speaking engagements such as today's session. Again, thanks for inviting me. Here's a overview of the four late 2021, from September to December to be exact, draft guidances for industry. On the left, we see assessing electronic health records and medical claims. We call this our EHR claims guidance. Right below that on the left, real-world data assessing registries. So we have data sources covered. On the right, data standards for drug and biologic submissions, since our regulations and data standards are based on clinical trial. And below that, considerations for the use of RWD and RWE to support regulatory decision-making, sort of an overarching guidance on regulatory considerations. To go into a little bit of detail, here again are the two guidances on sources of real-world data. So basically, to summarize, the EHR claims document gives sponsors, stakeholders, FDA's current thinking on considerations for selecting fit-for-use real-world data from those two types of data sources to help answer research questions of interest. Time doesn't allow me to go into a lot of detail, but the link is shown there. And in parallel, the registry's draft guidance offers considerations when designing or using an existing registry to support a regulatory decision. So again, I can't do much more than give a broad overview, but hopefully this puts the big picture into perspective. I did want to, though, scratch a little bit beneath the surface. Here's an example from the EHR claims guidance, and it's about what we've called, and others have called, the life cycle of EHR data. So the two excerpts on the left say the process for examining the quality of the data is not a one-time assessment. Rather, it is an ongoing process in multiple phases of the life cycle of spell check. Changed EHR to HER, as it often does, and I thought I found those instances, forgive me, but you get the idea. On the right, we have the figure that shows, I would focus on that column in various shades of gray. You go from source data at the bottom to curated data, to transformed data, to an analytic data set. That's clearly different from the shorter process involved with primary data collection in a clinical trial. So we want to be confident in the results, in the data that we see and the results that are generated. So again, this gives a little bit of a window into the EHR claims guidance. This slide focuses on the remaining two out of four draft guidances. The first one, data standards, recommendations for complying with the FD&C Act when submitting study data derived from real-world data sources, again, parentheses, versus a clinical trial, close parens, in an applicable regulatory submission. And the regulatory considerations are expectations for the design and conduct of non-interventional observational studies that are not subject to FDA's investigational new drug regulations, which is just a reality of the emergence of real-world evidence. To dig in again a little bit beneath the surface, for these two, I chose the regulatory considerations guidance, and I just have a copy. Well, it's not a quote, but they are copy paste, so these are verbatim. Regardless of a study's interventional or non-interventional design, the evidence submitted by a sponsor in a marketing application to support the safety and/or effectiveness of a drug must satisfy the applicable legal standards for the application to be approved or licensed. And the second one is, I'll try to paraphrase here, sponsors should engage the FDA early when designing a non-interventional study. For example, a Type C meeting could be requested with the appropriate review division to discuss expectations for the design and conduct, and sponsors should provide draft versions of their protocol and statistical analysis plan prior to finalizing these documents and before conducting the study analysis. Again, it should be apparent if one is doing a non-interventional study, that could be done after the fact, which raises all sorts of concerns. Whereas a clinical trial is done on a prospective basis, and besides randomization, there's that obvious added protection, if you will, from problems. Okay, so to put this all together, almost done with the guidances, we see the first 4 guidances that I talked about were published just as a heads up, and this information is also on our 2022 CDER guidance agenda, which lists what we're working on. We aspire to, by the end of the year, have an externally controlled trial guidance and an RCT and clinical practice settings guidance, which are currently in development. We hope to have them in clearance if not published, and those are on the design consideration side of the same landscape of how can FDA help by providing guidance. 2 other guidances. Here's the first of the 2. Just very briefly, digital health technologies for remote data acquisition and clinical investigations. If you don't mind me being informal, if data come from a wearable that I bought myself and someone figures out a way to pull that data, that would be real-world data, hopefully generating good real-world evidence. If you're running a trial and give me a wearable on my other wrist, on a strict technical basis, that is not real-world data because it was part of a clinical investigation. It might seem silly, but we do want to be consistent with our terminology. And bottom line is, this guidance gives recommendations for so-called DHTs in clinical investigations, and it's clinical investigations defined broadly as both trials and non-interventional studies. And here, a very recent guidance, submitting documents using RWD and RWE. This is a procedural guidance. It basically is asking you all to help us by being clearer in your cover letter in terms of what is being submitted, so that we could better track for internal purposes and also to honor a PDUFA 7 commitment to report on what real-world data, real-world evidence we're seeing. All right, now the fourth of 4 components of our program, I just have a couple of slides, but this will segue more to the 2 talks that preceded mine. First, let's start out very general. FDA supports RWE research projects to seed the field and hopefully improve the quality of either using real-world data or the quality of the data itself. In the middle column, figuratively and literally, you see a bucket of RWD study design, and on the right, we're calling it tools. So I'm going to actually, in the interest of time, not read all of these, but I'll let you look at them. And just, I know my next slide is an animation where I'm emphasizing 2 of them, not that the others aren't just as worthy, but as an example of using or improving RWD, we see a project to enable access to unstructured EHR data. And in the middle column, statistical methods for randomized controlled trial designs with hybrid control arms. One reason for the arrows being where they are is as a sort of a perpendicular look at these projects, we have different funding mechanisms that support us in our support of the field by seeding these projects. And this happened to be a so-called U01 award mechanism via Health and Human Services. The announcement was exploring the use of real-world data to generate real-world evidence in regulatory decision-making. And you see that we received 31 applications. They virtually all were quite strong, but it's a peer review process where the top 4 applications were funded. And I'm carrying the arrows from the prior slide to this slide, sort of in reverse order, where you see in the second row, the applicant was a combination of Genentech, University of North Carolina, and others who wanted to apply novel statistical approaches to develop a decision framework for hybrid RCT designs combining internal control arms with data from real-world data sources. The next row down, Variance as a private entity, got an award for transforming real-world evidence with unstructured and structured data to advance tailored therapy. And I want to now focus even more on the Genentech UNC, that second row, which is most relevant to today's discussion, by just pointing out, again, I read the title, so I won't repeat it in the upper portion of this slide, but the specific aims are: first, to evaluate hybrid trial designs and their operating characteristics with sequential monitoring and the use of adaptive randomization. Second, to assess possible extensions of the method beyond time to event settings when applied to diseases in different therapeutic areas. And third, to develop R packages supporting study design simulations and offering subsequent training workshops. So this is a project that, again, we're trying to, informally I'm using the phrase seed the field. I'm not endorsing it above and beyond any other project, nor am I endorsing it necessarily of its own accord. Time will tell what comes from this project, but as with all other projects, we are pleased to be in a position to be able to try to help improve where we see the need for greater clarity, and work in this space is being done by others who also deserve credit. They're just not FDA, linked to FDA, so I'm talking about what we are sort of, we let the project go on its own, but we are monitoring, we do engage with the proponents along the way. And here's the last slide in this project. This is an excerpt from the proposal. I'll just read it. Of special interest are hybrid randomized controlled trial designs, which supplement internal control arms with patient-level data from real-world data sources. Dissimilarity between internal and external controls has the potential to negatively impact the trial, such as decreased power, inflated type 1 error rate, as compared to RCTs. Bayesian methods, which adaptively adjust the influence of external controls on the analysis of trial data, can help to mitigate these issues and balance the risks and rewards associated with this type of complex trial design. So in effect, if I've made my point, this is one area where FDA is supporting an independent effort to try to help expand the field, and not just in the clinical topic areas that we're here today to talk about, but this is sort of a non-specialty specific, AKA non-denominational effort. And actually the trial they're looking at first is oncology. All right, now let's pivot to the next section. I just want to spend a couple of slides to sort of highlight what myself and Jacqueline Corrigan-Garay wrote about a few months back. Basically, the main issue is more than 5 years after passage of cures, the terms RWD and RWE are unfortunately being used inconsistently and interchangeably. I'll only spend the next slides talking about, I guess, the first two of the 5 contents of the article, adjusting 2 common misconceptions and providing a conceptual overview of study design if that helps. But I hope in general, just talking about this helps in a very broad sense. So the first misconception is that RWD and RWE are entirely new concepts. But as we said, in reality, sources of data and types of study design haven't fundamentally changed. But what has changed is electronic access to more detailed clinical data is evolving, and the data are becoming more relevant and reliable. The second misconception is that a simple dichotomy of randomized trials versus observational studies exists. But whereas in reality, clinical trials are defined by assignment of treatment according to a protocol. And if we think about it, single-arm trials face the same challenges as observational studies, perhaps slightly less, but similar in terms of determining whether the difference we see in clinical outcomes, in this case compared to an external control group, represents actual or true treatment effects. So what we proposed in a figure as shown here is focusing on terminology, but words matter. So randomized interventional studies is one category, non-randomized but still interventional studies is the second, and non-randomized non-interventional is the third. I won't go through this entire figure, but the next row down, still in bold font, you see that there are traditional trials which use real-world data in planning, but that real-world data, say, to assess trial feasibility or to support trial sites, trials always needed to do that. That is not generating real-world evidence. That's using data, period, to plan a trial, which we always had to do. So the generation of RWE starts with the second and goes to the third and the fourth of these 4 columns in the center of this figure. And again, trying to keep an eye on time. We have trials in clinical practice settings with pragmatic elements. Now that's actually, we have to be careful. Like pragmatic trial could have like 9 domains and be more or less pragmatic. But what we're trying to say there is certainly if a point of care trial, for example, collects the outcome from a real-world data source, say from the electronic health record, that is using real-world data in a substantive way to generate real-world evidence. Moving to the right, an externally controlled trial where we have a single-arm trial with an external control group from real-world data. I think everyone would agree, although, to be fair, right, externally controlled trials were done prior to 21st century cures. They're in our regs as historical controlled trials from the 70s. But nonetheless, in contemporary speak, it is real-world evidence and, of course, observational studies. So there's an increased reliance on RWD, as the arrow shows, as you go from left to right. But this can hopefully, this conceptual approach can hopefully help. But along those same lines, as I was just saying, this slide shows how real-world evidence informs effectiveness when fit for purpose from before 21st century cures. I'll just look at the third row, blinatumomab, Blincyto, a single-arm trial with an external control group that was approved in 2014. Here, I'll go to the fourth row. This is during the 21st century cures era. We see Zolgensma. Again, it happens to be a single-arm trial with a natural history comparator arm, but we get the idea. So what I want to do in wrapping up is just say, how does FDA approach this situation? Our key consideration, sort of a review of systems, is whether the RWD are fit for use, whether the trial or study design used to generate RWE can provide adequate scientific evidence, and whether the study conduct meets FDA regulatory requirements. And I know this is very big picture level, very high level, but we operationalize this. I'm running short on time, and this is about a year old, but I do want to mention this is not a trial, but this is the approval, since we're talking about transplant of, as we know, tacrolimus for lung, where it hadn't been approved other than for, well, it had been approved, excuse me, for liver, kidney, and heart, but it had not been approved for lung. The sponsor submitted a non-interventional, quote-unquote, real-world evidence study. And next, this slide shows what I referred to on a prior slide about the data, the design, and the conduct. Basically, the data were fit for purpose. They were as good as a trial would have collected. The non-interventional treatment arm was compared to historical controls, and the patient-level data were provided. This is very cursory, but accurate. So we determined this to be well adequate and well controlled. And of course, the fact that organ rejection and death are virtually certain without therapy, and these dramatic effects of treatment helps to preclude bias as an explanation of results. If that sounds like too good to be true, it's not. It happened, but here's the flip side. If that sounds like hard to emulate, it's I guess hard to replicate. We see problems with real-world data sources. We see problems with non-randomized study designs and the conduct of non-randomized studies. These slides will be available, so in the interest of time, I'll let you read this, glance at it now and read it later. But we are asking you to consider these issues before you submit to FDA. And as I wrap up, I'll just say that here's a closing paragraph from that article. We remain committed to robust policy development aligned with the 21st Century Cures Act, while we maintain our evidentiary standards and honoring our obligation to protect and promote public health. So if we all focus on the distinction between interventional and non-interventional studies and other terminology, we can help researchers, sponsors, and regulators better understand and describe relevant methodologic issues before we get to the case-by-case assessment of whether or not the data are substantial evidence. And certainly gaining more experience, including the conduct of rigorous non-interventional or trials with hybrid control arms, will help us to advance drug development. With that, I'll say thank you very much. And if there are any questions after this session, the email address shown here can be used to get in touch with me or to get in touch with our program in general. Thanks again for having me.
Okay. Hello, everybody. So this is panel discussion 2, focused on real-world evidence. I'm Amanda Klein, the Executive Director for the Transplant Therapeutics Consortium. With me is Luke Kosinski, who is a quantitative medicine scientist for the Critical Path Institute, Alice Toll, Research Analyst for UNOS, and John Concato, Director for Real-World Evidence at FDA. So there's been a couple of questions in the chat. For the first question, this one is going to be directed towards Luke. The question is, in the example with real-world evidence supplementation of graft survival at 5 years, it seems we would still not reach over 1,000 recipients due to low propensity matching. Can you elaborate and provide feedback or comment?
Sure. So, for a future study, we should actually have more patients available, because always accumulating patients. My understanding is we should be getting about like 25,000 or maybe more patients per year. So for use in a future, planned trial, we should be able to get 1,000 patients without trouble.
Okay, thank you. And Alice, was there anything you wanted to piggyback with that for those who are less familiar with the UNOS registry?
Yeah, so in the analysis I was showing, we were limited to a 1.5-year time frame. So, we could expand that time frame. We were really trying to match what had been done in the benefit trial previously. And so if you were planning on using real world data, you would, maybe set up your inclusion and exclusion criteria differently to meet your targeted number of controls.
Thank you, Alice.
And this is John. May I comment, Amanda?
John. Go for it.
Yeah, no, just I'm struck by the complexity here, right? On the one hand, we have it's not that they're mutually exclusive, but we have the capital S statistical issues of, how are we going to incorporate these real world data controls into a hybrid comparator arm? And then the methodologic side or the clinical methodologic side is, if you have to go back farther in time, and I think there's a question or a comment in the chat, we start to get nervous because the standard of care can change. It's context specific, but we have to keep track of all of these different aspects, including, just as I think the first comment implied, if it's not as simple as a percentage, but if you have 99% real world data controls and 1% in the trial, I'm exaggerating to make the obvious point, that doesn't feel like a clinical trial anymore. If it's the converse, if it's 99% RCT, then we might say, well, why do we need the 1%? And it's not as simple as finding a balance in the middle. But I think we just have to keep track of these different aspects of the design and be thoughtful. And of course, it ends up being a review issue at the end of the day, but we want to have enough clarity going forward step by step to increase the likelihood of finding useful therapies going forward on behalf of our patients.
Thank you, John. And just to echo, what's unique about the UNOS registry in the United States is that it captures all transplant recipients. So, unlike different therapeutics, right, we may not know who has diabetes, for example. They may not even know. But with transplant, we have the unique opportunity to leverage real-world evidence because we have the entire population of the United States captured, and right. that uniquely qualified to explore this. So thank you for all that comments.
Yeah. I was worried about having not enough time for my talk, but now it looks like we have time. I just want to echo that. I think I said in the pre-recording that clearly the data were viewed as high quality, and that's representative of registries in general, right? It really is data collection according to a protocol, even if the treatment of interest isn't assigned. But when I give talks and I mention ProGraph to other audiences, I also talk about the generalizability. It doesn't get any better than having all the transplants in the United States. And that is rarely, if ever, the case, say, with electronic health record data. So I just want to emphasize the point you made, Amanda. Sorry for belaboring, but I think it is important for the audience to hear.
I agree. Thank you. This question is for Alice. Are you able to do this pilot study for real-world evidence with thymo, tacrolimus, mycophenolate, prednisone as the control arm? And really, I think the background with that is that's the current standard of care in transplant medicine to compare to the Simulect, belatacept, mycophenolate, prednisone. And can you comment on that?
Yeah, so we didn't look at thymo because it wasn't standard care while the original benefit trial was going on, and we were matching our cohorts based on time period. But if you wanted to do some sort of analysis where you matched on a more recent standard of care control cohort, that's a possibility. We collect many different immunosuppression regimens in the OPTN data, so kind of the combinations there is endless.
All right. Thank you, Alice. And I think that the idea of this pilot study was to test it, right, to see if we can use external control arms from registry data and compare to a completed RCT. So this is really the initial exploration. So we have time for one more question. Darren Stewart, I think this one could be for John. Is there consensus on whether external real-world evidence controls should be contemporaneous versus historical, given concerns about crossover, contamination, and other considerations?
Yes, thank you for the question, and I was partially addressing that, I think, in my earlier comments. But I would also say, referring to an earlier slide where I mentioned that on our 2022 CEDAR guidance agenda, which lists guidance that are in preparation, one is on externally controlled trials, which again, what we've talked about today is a quote unquote hybrid RCT, but there are parallels, right? We're taking the real world data sources and adding them in as part of the control group. And I would say that I'll use an imprecise figure, approximately 10 different domains of comparability that we worry about. And certainly one of them is historical comparability. Again, as I mentioned, the further you go back, the more treatments change. In some clinical contexts, it's more stable than others, but it's a case-by-case basis. So ideally, we would have geographic comparability, we would have historical comparability, we would have prognostic comparability, which the propensity score is attempting to get at, and I could go on and on. So I think we have to look at this in a very rigorous and structured way to determine when are we still confident in the data. In part, I must say, again, ProGraph, we had the historical control with 100% outcome. And as those who work in the field know, for the most common regimen, the 1 year outcome was only 9%. So that difference, that delta, is so strong that it really gives us high confidence that bias is not likely to explain the results. It's fantastic that 5-year survival is as high as it is, or any outcome, graft survival or overall survival, and we're trying to do better. But if we're talking about some of the numbers that Luke, you were showing earlier with the GUI, right? And we're talking about between a 3 to a 5% delta. That's where, as you'd imagine, I'm not speaking for the review teams nor the statisticians at FDA, but as an internist and epidemiologist, one gets less comfortable saying that we're excluding bias or confounding. And as Alice nicely described, the data sets are small. We do the best we can. And even if the standardized differences, right, Alice, were 0, I mean, we'd have high confidence with the variables we have, but that doesn't mean that some variables that we don't have might not be in balance. So it's a complex situation, but we stand ready to, again, evaluate the potential use. This is, I would say, we're using real-world data here. We generate real-world evidence when we do the hybrid trial, but we're pulling in real-world data on a strict terminology basis for a lot of this discussion today. I hope that helps, Amanda and everyone.
It does. I think it nicely summarizes the importance of integrating the regulatory considerations, the design considerations, the clinical considerations, that everyone has confidence in the output. So thank you for highlighting those points. So with that, we're going to end the panel discussion and move towards session 3, which is focused on the future of transplantation, kidney transplantation. So thank you all.
Well, I want to thank the organizers for the opportunity to come and talk about efficacy endpoints in kidney transplantation. A couple disclosures: I have some consultancies and honoraria. I will not talk about off-label or investigational use of drugs. So the objectives for this talk are to discuss the causal pathways leading to all-cause kidney allograft loss, discuss the evolution of some trial designs in kidney transplantation, discuss the actual efficacy of modern immunosuppression and what we've been learning recently, and applying that then into future efficacy endpoints in kidney transplant trials. And I'm going to also speak a little bit to the prognostic biomarkers as tools to optimize the use of these efficacy endpoints near the end of the talk. So we have seen progressive improvement in patient and kidney graft outcomes for both deceased and living donors, both in terms of how the patients are improving their outcomes, but also in terms of graft survival. Unfortunately, it's still pretty clear that even in the most recent data in 2012, 2015 outcomes for deceased and living donor, we're still losing a lot of our grafts by 5 to 10 years post-transplant. So what are today's barriers? Well, if we look at it from a causal pathway point of view, ultimately we have graft failure and death at the end, but graft failure is preceded generally by loss of organ function. And this is related to scarring either in the interstitial compartment or in the filters of the kidney, as it's called glomerulopathy. We can have recurrent disease that leads to glomerulopathy, and we see that not infrequently. But most importantly, we have the immune response to the graft, which is triggered by the HLA disparities between the donor and the recipient. Some recipients have preformed memory, and so when they get challenged with that same HLA mismatch, it can trigger off a T-cell mediated rejection or a antibody mediated rejection. Or the mismatch itself at the time of transplant is a strong drive to lead to TCMR or T-cell mediated rejection, be it subclinical or clinical, or the development of a donor-specific antibody or DSA leading to antibody-mediated rejection. And these rejections can continue on in what are called chronic active or smoldering rejections that ultimately lead to scarring and fibrosis inside the kidney. Now, if you have an older donor, those organs tend to repair less to injury. And the ischemia reperfusion injury, which occurs with any transplant, especially with deceased donor transplants, can lead to delayed graft function, which itself can lead to an injury response with formation of scarring as the healing modality. Now, what we do is we give immunosuppressive drugs to try and suppress and limit the impact of ischemia reperfusion injury and the degree of mismatch between the donor and the recipient. Unfortunately, these drugs come with side effects, and it leads to metabolic dysfunction, infection, and cancer on a background of comorbidities that the patients already have. Or they can be directly toxic in terms of being calcineurin-induced toxicity, leading to injury to the graft, or leading to infections that themselves are directly injurious to the graft, like BK virus nephropathy. Now, as these side effects and toxicities arise, our response is typically to minimize their immunosuppression, or the patients not liking the side effects themselves stop taking the medications. Either of these, physician-guided or patient-led, leads to a reduction in effective immunosuppression that can then lead to immune allograft rejection. So the future for us is really to come up with novel drugs that have improved efficacy and less toxicity as compared to what we're currently using. So how have we evolved in our trial designs, and what endpoints have we been doing for drug development in transplant? Well, the traditional designs in phase 2 and 3 have been taking a test drug versus an active control. And we've been looking at the primary endpoint being patient graft survival, and later incorporating biopsy-proven acute rejection, or BPAR, efficacy failure. Now, BPAR efficacy failure is a composite definition used by the FDA as all biopsy-proven acute rejection events, as well as graft failure, death, or loss to follow-up in those subjects without BPAR. And BPAR efficacy failure has become really the primary endpoint in RCTs for the evaluation of efficacy and for the label indication of prophylaxis of rejection in kidney transplant recipients. If we look at the evolution of drugs that have been approved in transplantation, you can see early on it was more case series as opposed to formal clinical trials with the use of Imuran. It was developed by Roy Kahn and Murray in Boston. And subsequently, as we went to calcineurin inhibitors in the 1980s, we saw improved graft survival, patient survival, death-censored graft survival, and decreased acute rejection, although it did come at a cost of some impaired renal function related to CNI toxicity. And as we've moved up, we no longer could show short-term benefits in terms of patient and graft survival, but we saw, again, progressive decrease in acute rejection that was found on a for-cause biopsy, with some improvement in kidney function. That got us through MMF, and ultimately we got to tacrolimus, which is one of our current drugs that we're using that replaced cyclosporin because it was decreasing the acute rejection rates as compared to cyclosporin. As we got up into the 2000s, we no longer could show even superiority in terms of acute rejection. We could show equivalence, and that's how everolimus was approved in a equivalence trial with some impairment at the same time in graft function. The last drug to be approved in 2011, belatacept, which I think you're going to hear more about or have heard more about today, was a non-inferiority trial where in the short term we showed equivalence in terms of non-inferiority. Even though there was a slight increase in acute rejection, there was improvement in the secondary endpoint of improved graft function. And this is where we've ended up with non-inferiority designs. So what's important about a non-inferiority trial is to show a new drug is not materially worse than the current standard of care or control. And it's reasonable to consider a non-inferiority trial if the investigational offers clear benefits, maybe not in terms of efficacy, but in terms of other toxicities or side effect profiles. It does assume superiority of your standard of care versus a historical placebo control to define what is called a pre-specified non-inferiority margin, or M1. And it must be based on evidence. And it must, the current trial that you're going to use should mirror some of the historical trials. So to give you a sense of what a non-inferiority margin it looks like for BPAR efficacy failure, the FDA used this studies of cyclosporin MMF as steroids, where they derived a point estimate of BPAR of 23.5%, and they compared that to the BPAR efficacy failure that occurred on an MMF or steroid in a, as they called it, the placebo arm. And here you could see that the point estimate was 58%. And when they calculated their M1, they came up with an M1 of 22%, taking the upper bound of the active control versus the lower bound of placebo. Now, the FDA guidelines suggest that we should really be setting an F2, which is a clinically acceptable loss of efficacy, being less than 50% of M1. But that has, you could go higher than that if it was justified. So how do we put this into interpreting a trial? Well, if you have non-inferiority demonstrated because the upper bound here is way less than M1, it's actually on the other side of 0. Here you can say it's not only non-inferiority demonstrated, but it's actually superior. If the point estimate is 0 and your upper bound is less than M1, you can say that it may be equivalent. In this case, in green, what I'm showing you is the point estimate is tending to favor the control drug, but the upper bound is less than the M2, that was the clinically acceptable loss of efficacy in this case, as compared to this case where the point estimate is also greater than 0, but now the upper bound crosses the M2. And this would be interpreted as non-inferiority demonstrated, but it's a clinically unacceptable loss of efficacy. If we get into this black scenario, we see that it's again non-inferior because the upper bound is less than M1, but actually because the lower bound is greater than 0, this is actually inferior to control. And in the orange example, what I'm showing you is non-inferiority has not been demonstrated because the upper bound now is actually above M1. So that's how you would interpret a non-inferiority trial. And the ways to win in a trial design would be to hit one of these 3 scenarios in blue or green. So now let's look at the efficacy of therapeutics today in terms of suppressing alloimmunity and improving graft survival. And more recently, we've conducted a trial in Manitoba looking at a cohort of patients who were only on what is called standard of care, TAC, MMF, and prednisone. They had no pre-existing memory. We ruled out this by absence of DSA using single antigen B testing at the time of transplant. And what I'm showing you is all the for-cause biopsies we did in this cohort of 775 individuals out to the first 5 years. And then I'm also showing you where we did surveillance biopsies. And what's interesting here is if we use borderline rejection or higher as the definition of rejection, by 1 year, we're at a BPAR rate of about 22.5%. Now, this is much higher than that reported in most literature today of claiming a BPAR efficacy failure rate on just for-cause biopsies alone of around 5 to 6%. So what I'm showing you here is that actually BPAR is more common than we think when you start including the use of surveillance biopsies. In fact, in our study, what we were able to show is that a TCMR or an ABMR event occurred in 30% of the individuals in this cohort, despite standard of care therapy. And more interestingly, and something that has not been well reported in the past, is that those patients who had a first TCMR and were treated for it with predominantly steroid therapy, a full 50% had a second TCMR event. Now, this is interesting because this has been not appreciated that we have that high a failure rate in the use of our anti-rejection therapeutics. Now, if we look at the biopsy grades of rejection, looking at comparing cyclosporin and FK-based therapy, What the big advantage of FK is it actually decreased all the higher grades of rejection in red here as compared to cyclosporin. And it also decreased the rate of borderline, although this still now is under tacrolimus-based therapy, the most prevalent form of T-cell mediated rejection. And now fully 70% of patients have no rejection in that first year as compared to cyclosporin. So what we've effectively done with tacrolimus-based therapy is shift the grades of rejection downwards, but we still have this borderline category. And when we look at the study I just showed you in the surveillance or for-cause biopsy, the most common diagnosis was borderline rejection on the for-cause or surveillance biopsy. And on the for-cause biopsies, there was an increased prevalence of the other grades of rejection as compared to on the surveillance biopsies. Now, in 1993, the Banff classification set the threshold between borderline and grade 1 to define acute rejection. So they were calling borderline as suspicious for rejection, but they weren't fully calling it rejection. They set this threshold and they designed it artificially so that the false positive rate in the diagnosis of rejection should be very low. And that was reasonable in the days of cyclosporin and Imuran. They didn't want to be over-calling rejection. But that has the potential for frequent false negatives, depending on the prevalence and impact of Banff borderline TCMR. And as I've already shown you, on TAC MPA, which is the current standard of care, that's the most prevalent diagnosis you're going to see on a for-cause or a protocol biopsy. Now, we went and looked in a time-dependent covariate analysis to see whether or not the first TCMR was actually prognostic of the death-censored graft loss or all-cause graft loss. And what we were able to show was that DGF, first TCMRs, and antibody-mediated rejections were all independent correlates of death-censored and all-cause graft loss. We did a sensitivity analysis to ask whether it mattered whether the first TCMR was found on a for-cause biopsy or on a surveillance biopsy. And what we found was that absolutely on a for-cause biopsy, it was a marked increased hazard for death-censored and all-cause graft loss. But even on a surveillance biopsy, our hazard ratio was almost twofold, even though it didn't quite get to significance. And I think this was largely because of the number of events we had, rather than it really not being a significant outcome. And this was all independent of antibody-mediated rejection. And I'm emphasizing this because a lot of the literature has been arguing that only antibody-mediated rejection now matters and that T-cell mediated rejection is not as important. This data would refute that the independent of antibody-mediated rejection, TCMR is still an important outcome for our patients. When we looked at the grade of TCMR, we found that a Banff borderline rejection and a Banff greater than or equal to 1A each had a very marked hazard for developing death-censored graft loss. And for all-cause graft loss, it was really the Banff greater than 1A. So borderline rejection does matter in terms of having an impact on the current standard of care and suggesting we should really be considering borderline as in the rejection phenotype. And in fact, there's other literature now coming across the world suggesting that borderline has long-term implications for our patients. The second thing I want to emphasize, again, a study that we had done in our center, was a systematic review and meta-analysis of if the initial TCMR was greater than or equal to Banff borderline, what was the incidence of a second TCMR occurring in these patients? And we can see that the point estimate is 39% of the patients, with a range of 26 to 53, were having second events. And this is telling us that a second TCMR is actually quite common. That was true whether it was Banff borderline to start with or it was Banff 1A to start with. We got the same point estimate. So persistent TCMR in the following 2 to 9 months after anti-rejection therapy is actually as common as what we saw in our own cohort. So what's the impact of these second TCMRs? Well, again, doing a time-dependent covariate analysis, we saw that the second TCMR was actually almost a 3-fold hazard of developing death-censored graft loss and a 2.3 hazard of developing all-cause graft loss, independent, again, of ABMR or delayed graft function, telling us that this persistent inflammation is absolutely poor for the graft. And again, we did a sensitivity analysis to look at whether that second TCMR was occurring very quickly after the first one, or if it was arising again in a longer time period after 6 months. Didn't really matter. Both were significant for both death-censored and all-cause graft loss. So this has really gotten the community interested in the concept of how do we detect these rejections non-invasively through diagnostic tools, be it blood mRNA, urine mRNA, or urine proteins. And there's a lot of different papers that have come out. This is a review and highlighting that there's different specific molecules, but most commonly nowadays, it's a multi-gene signature that people are using. Either there's a blood mRNA, urine mRNA, or actual urine proteins. So we're getting into this whole concept that maybe we don't need a biopsy to diagnose rejection. We could get into non-invasive tools. So this brings me to the first part here, which is, what's the status of efficacy endpoints? Well, patient and graft survival, superiority is not feasible in a short-term phase 3 trial. You're going to have 5 years or more of follow-up. Non-inferiority cannot be done for patient and graft survival because we cannot determine a placebo effect, and it's not possible to define this, therefore, an evidence-based non-inferiority margin. BPAR efficacy failure, as defined by the FDA as BPAR graft loss, death, or loss to follow-up, I think superiority is still possible, especially if we consider a biomarker enrichment strategy, which we're going to talk about in a minute. And non-inferiority, while it requires this non-inferiority margin with a clinically acceptable M2 for regulatory approval, I think we can still do non-inferiority trials where if we were to employ a biomarker stratification strategy with some adaptive enrichment, which we'll come to in a minute. Newer things on the horizon, renal function endpoints. I think this is a near-term opportunity. Certainly in chronic kidney disease trials, the FDA will consider it as an efficacy endpoint, looking at 2 to 3 years, EGFR declines of greater than 30 or 40%, versus the EGFR slope, where you've lost greater than 0.5 to a mil per minute per 1.73 meters squared per year. Also looking at urine albumin creatinine ratios having a greater than 30% change. So these are tools that are coming into the general CKD trial designs, and I think we are likely to see those evolve into transplantation. The other opportunity is non-invasive diagnostics for the detection of rejection. I think this is a more future opportunity. They can be non-specific, things like serum cell-free donor-derived DNA, the urine or blood mRNA profiles I was talking about, or urine proteins, or a more specific diagnostic like a donor-specific HLA antibody. All of these being used to detect rejection where we wouldn't necessarily need a surveillance biopsy as the endpoint. So where do I think the real opportunity exists? I still think it's in considering BPAR efficacy failure as the key efficacy endpoint, but it may be how we define it and how we use it in our trial designs. So here I want to talk a little bit about prognostic biomarkers in trial designs. And one of the points I want to highlight here is that really immunosuppression is escalated to the highest level tolerated. We're giving TAC-MPA to almost everybody, almost 2/3 get steroids. We follow creatinine and TAC, and we adjust our immunosuppression in response to rejection, infection, or toxicities. But we all know that there's a heterogeneous treatment effect of the drugs, and the challenge for us is to get the right therapy to the right recipient. And certainly all of our trials to date have really not been trying to do that. So what we really need is a way of having alloimmune risk assessment, either assessing for memory, which we've been doing for many years, or assessing for the risk of a primary immune response, which would be highly relevant in our trial designs. And we've been thinking about this for many years around HLA mismatch, around class 2 molecules like HLA-DR, race, and recipient age. Unfortunately, if you actually look at how physicians practice, very little of the variation in the utilization of our drug therapy is based on the risk profile, less than 11% for induction therapy and less than 5% for maintenance immunosuppression. Most of the variation is based on how physicians practice at that center. So telling us that they really don't invest a lot in their alloimmune risk assessment as part of their decision-making for the drugs that they're using. However, what we've been working on is coming up with a HLA-DR-DQ molecular mismatch score. And we've shown that that's a prognostic biomarker for primary alloimmunity in kidney transplantation, such that we can look at prognosticating 3 levels of risk for developing a de novo donor-specific HLA antibody, low versus intermediate versus high risk. And this partitions the entire general transplant cohort into 25, 35, and 40% of all transplants that are being done. This translates into the risk for antibody-mediated rejection over the first 10 years. More importantly, it actually translates into the risk for developing TCMR in the first 12 months after the transplant, low, intermediate, and high. Not only does it prognosticate the risk of developing a TCMR, it also prognosticates the grade of TCMR. The patients that are getting the higher grades tend to be in the high risk group, and the ones that are having more recurrent rejections also tend to be in the high molecular mismatch group. So we've submitted this to the FDA under their biomarker qualification program to consider a prognostic biomarker that could categorize patients at the high, intermediate, and low risk for developing DSA rejection graft failure, such that we could use these categories for enrichment or in stratification design of phase 2 and 3 trials. The next step, which is already underway, is clinical trials to support our qualification plan. So then I want you to then consider, what is it we're trying to do in novel drug development in kidney transplant? Well, if you think about the molecular mismatch, the high-risk mismatch are the ones that need a more efficacious drug. That's their priority. And if we look at who those individuals are, and I'm showing you a plot here, the risk of a first TCMR goes down dramatically as you get older, such that by the time you're 50, you're at a nadir for the risk of developing a TCMR. These are residual plots from our data. And that makes sense. A lot of what we're doing in vaccination strategy around COVID, we know that the elderly have poorer responses to vaccine. We know that the younger people have better responses. Nothing different here when we do a transplant. We're immunizing with HLA molecules, and it's resulting in rejection. So the patients that need the most improvement in efficacy are going to be your patients under the age of 50, where they have a high molecular mismatch. So this is the quadrant that really needs a more efficacious drug. If you think about the patients who are over 50, their biggest problem is that they have all-cause graft loss, which is really another way of saying these individuals are dying with their comorbidities and their toxicities related to the drugs. So the priority of an older individual, especially if they have a low molecular mismatch, should be on minimizing their toxicity. These are two different competing priorities. One's on improving efficacy. The other is maintaining efficacy while minimizing toxicity. So what could a design look like? Well, you could have a phase 2-3 design where you're doing enrichment based on the molecular mismatch score, and your priority here is on efficacy, and your superiority of your primary endpoint is going to again be on BPAR efficacy failure. But now that efficacy endpoint should be considering some of these newer definitions of BPAR, like borderline TCMR, the use of surveillance biopsies to diagnose subclinical rejection that's going on in the graft that you didn't know was apparent. Or ultimately, we'd like to get into the defining of BPAR efficacy failure using not an invasive biopsy, but non-invasive diagnostics. Another design would be to stratify by molecular mismatch, and where your priority here is on decreasing toxicity. It's a non-inferiority design using, again, your primary endpoint as BPAR efficacy failure, using the same definitions we just talked about as modified and given the current context. But here you could do adaptive enrichment, because what you're trying to do is give a drug that's equally efficacious or marginally less efficacious, but it's got a big benefit in terms of its toxicity. So your secondary endpoints would be improved renal function, improved cognition, less diabetes, less infections, less tumors. This is the things that we see with our current standard of care. So here with adaptive enrichment, you could look, and it may be certain strata really tolerate being on an equally or slightly less efficacious drug. So they maintain their efficacy in that strata, and they have improvement in their secondary endpoints. The other unmet need, I think it's apparent from the data that I was showing you earlier, is that clinical trials for the treatment, not prophylaxis, but the treatment of TCMR to improve long-term outcomes have really been ignored. So we have early TCMR that can lead to persistent TCMR, the development of DSA, and both of these events can lead to chronic active ABMR and TCMR, which leads to reduced graft failure. What we'd really like is a more efficacious drug that leads to more remission of the TCMR with no graft inflammation to have a better outcome. So to that end, we've actually formed a working group called the International T-Cell Mediated Rejection Working Group. The sponsors are the AST, ESOT, TTS, and the BAM Foundation for Allograft Pathology. We have subject matter experts, regulators, and patients from AAKP and the European Kidney Patients Federation. And our goal here is to develop clinical trial designs to evaluate novel therapeutics for the treatment of TCMR. And I highlight this because the last drug that was approved for the treatment of TCMR was in 1998. So we know we have a very high post-treatment event rate on the current standard of care. There's every reason to think we could do better than that. And I think this is a real low-hanging fruit for us to reinvest in, and using efficacy outcomes being your resolving of the TCMR, which right now we know that there's a 39% failure rate. So to summarize, BPAR and drug toxicity are limiting the effectiveness of our current standard of care. I think BPAR-based superiority and non-inferiority trial designs remain viable today. What is the evolution is the definition of how we define BPAR efficacy failure. I think we need to think about including borderline within the definition of TCMR. That would give us a higher prevalence rate. We need surveillance biopsies to detect these TCMRs and ABMRs that are not quite causing graft dysfunction, but we know left untreated can lead to poor graft outcomes. And ideally, we develop non-invasive diagnostics as part of our way of defining BPAR efficacy failure for detection of both TCMR and ABMR. But I think that's a bit more of a future opportunity. We need the development of renal functions and efficacy endpoint. I think this is coming, whether it's going to be eGFR decline by a certain percentage versus eGFR slope. I think, again, we will find out in the days to come, but I think this is on the horizon and doable, working with the regulators and presenting the right data. And then I think we need targeted trial design utilizing enrichment or stratification, and the HLA molecular mismatch tool is an example of how we could enrich in our superiority trials to keep our sample sizes smaller, or stratify in non-inferiority designs to find out whether there's subgroups that benefit from an introduction of a drug that may not be any more or might be slightly less efficacious, but it certainly would allow an improved toxicity profile. And with that, I'm going to stop and just acknowledge my many collaborators that have been involved with a lot of the work and thought that went into this talk. Thank you very much.
I'm Bill Fitzsimmons, and it's my pleasure to present today on safety endpoints in kidney transplantation. Previously in this webinar, you've heard a fair amount about efficacy endpoints, including the novel Ibox scoring system from Amanda Klein, and some of the new future potential endpoints from Peter Nickerson. I would like to now move to the other side of the benefit-risk equation and speak about safety endpoints, because I believe that this is an area that can also be important as we all try to improve the outcomes long-term in kidney transplant recipients, and also try to facilitate innovation to bring new immunosuppressants into this field. The following are my disclosures. And as I start, I would like for you to think of the answer to this question. In terms of the needs for innovation in de novo kidney transplant immunosuppression, which is a greater need? Do we need more efficacious immunosuppressants, or do we need safer immunosuppressants? And this may be a bit of a trick question in that the true ideal answer is we need both, and hopefully both in the same therapy. But that may be a very tall order and very difficult to achieve. So what I would like to do is hopefully make you think more about developing safer immunosuppressants by using safety endpoints in the trials, And this will be a great topic for our subsequent panel discussion. So first, I'd like to say why I think focusing on safety endpoints is important. And I think there's 4 main reasons for doing that, and I'll provide some of the data behind these reasons. But the first and probably the most important is that as we look to improve long-term outcomes in kidney transplant recipients, safety events and adverse events of our immunosuppression have impact on the outcomes of death and graft loss. In particular, if we look at first that overall outcome of the combined graft loss, meaning death with graft function or graft failure, there's a recent paper that came from Merzkani and others at the 3 Mayo Clinic centers with a senior author of Mark Stegall, which showed the data on 5,752 kidney transplants performed at the 3 centers. And on the left side, if you can see what all of us are trying to improve upon, the black line is the overall graft loss, meaning death with function or graft failure over time post-transplant. And the 2 main components of that being graft failure in green or death with a functioning graft in red. And as you can see, those 2 components are virtually identical for approximately the first 4 years post-transplant. But beyond that, they start to diverge over time, with death with a functioning graft being the larger contributor to the overall graft loss incidence. And if you look at the right side, table 2 from the publication, you can see the top reasons for death with a functioning graft, that being malignancy, infection, and cardiac causes, with malignancy tending to be a longer-term endpoint, increasing in incidence as you go beyond 1 year post-transplant, infection being more short-term focused with a decreasing incidence over time post-transplant beyond 1 year, and cardiac reasons being fairly stable. Similar to this experience at Mayo Clinic, if we look at the overall US RDS data from the years from 1994 to 2000 in terms of the transplant years, you can see that those top 3 reasons for death with a functioning graft remain. That being number 1, cardiovascular disease, 2, infections, and 3, tumors. So there's very consistent data that we know the 3 primary causes of death with a functioning graft, which are the largest causes of late graft failure and graft loss in kidney transplant recipients. If we then turn to the other side of that and look at specifically at death-censored graft loss or graft failure, the University of Wisconsin published a histopathologic analysis of the reasons for death-censored graft loss. And you can see that the top reasons, the top 3 reasons here, are all alloimmune response to the graft, that being acute rejection, which tended to be in the first 5 to 6 years post-transplant, transplant glomerulopathy, which was longer beyond 6 years post-transplant, interstitial fibrosis and tubular atrophy as well. But beyond the alloimmune causes, recurrence of disease and BK nephropathy stood out also as the top reasons for death-censored graft loss. If you compare that to the Mayo Clinic experience in over 5,000 kidney transplant recipients, you see their graft failure rates here and the reasons for that. The number 1 reason being alloimmune, again, consistent with the data from the University of Wisconsin. Secondly, glomerular diseases or recurrence. And third is renal tubular injuries or nephrotoxicity, potentially related to the immunosuppressive therapies. And also included in this list, BK nephropathy, which we also know is associated with the intensity of immunosuppression and particularly higher in certain immunosuppressive regimens like TAC plus MMF. I thought that as I reviewed the Mayo Clinic publication, there was a very important quote in the discussion section where the authors indicate that if you count both the deaths with a functioning graft as well as graft failure, less than 25% of the overall graft losses in their study were due to alloimmunity. And therefore, attempts to reduce the incidence of mediated graft loss, although it may be important in the younger patients, is unlikely to significantly change outcomes in the population that are most commonly transplanted today. And I think that that's important in that oftentimes we discuss more unique and novel mechanisms for immunosuppression and the need for better efficacy. And certainly that will improve some of the patients, but it looks like it's a minority of the patients and a minority of the overall graft losses that will be impacting even with more efficacious immunosuppressive therapies. And I believe that that's consistent with what you'd seen previously presented by Peter Nickerson, who was kind enough to share this slide with me. As we focus on more efficacious immunosuppressants, that may impact the upper left-hand corner of the patient segments, those who have a higher risk of TCMR and subsequent alloimmune graft loss, who tend to be younger patients with higher eplet mismatches. But that may only be 25, 30% of our overall population, whereas we also need to think about the patients that are in the blue, as well as in the lower right-hand corner, those who are in the red, where the risk of rejection may be particularly low, but the risk of toxicity and their overall death with a functioning graft and other reasons for graft loss particularly high. And you can see that in the graphs on the far right, where the first TCMR rate drops and plateaus and somewhat as you get beyond 40 years of age for the recipient. But as you get above 40 to 50 years of age for the recipient, the all-cause graft loss goes up. So to summarize that, if we look at the contribution of immunosuppression to the hard outcomes of death or graft loss, again, the consistent causes of death with a functioning graft, cardiovascular, particularly stroke, heart disease, and diabetes related, infection and cancer, all are impacted, and immunosuppression contributes to those three top causes. In addition, if we look at the causes of death sensor graft loss, clearly BK nephropathy and nephrotoxicity or renal tubular injuries are impacted by immunosuppression. But I'd like to make the case that adverse events of immunosuppression also contribute to alloimmune rejection. So I believe that part of the rejection process can be a result of the adverse events of the immunosuppression. Now, why would that be? The way that I would like to think about that is that there are direct and indirect effects of these adverse events. The obvious direct effects would be things such as diabetes, where we know that if we increase the incidence of new onset of diabetes after transplant or worsening diabetes, that contributes to an increased risk of death and graft loss. That's a direct effect of diabetes. On the other hand, if we look at an indirect effect of adverse events, when patients experience adverse events, oftentimes the immunosuppressive regimen is modified or minimized, reduced by the healthcare professionals, or the patients may become non-adherent to their immunosuppressive regimen due to adverse events. The result of that is we could end up with suboptimal immunosuppression, which then contributes to alloimmune graft loss. And I think we've seen some of the most extreme cases of this approach for instance, the CNI avoidance protocols historically done in the transplant field, where problems in terms of alloimmune graft loss occurred due to suboptimal immunosuppression, that immunosuppression did improve adverse events, but it also resulted in an overwhelmingly higher rate of immune-mediated injury to the graft. So you may say, how big of an issue is the indirect effect? If we look at the phase 3 trials that were key to the registration of the three of the most recent immunosuppressants, that being Nulojix or belatacept, or the 2 extended release tacrolimus formulations of Envarsus XR and Astrograph XL. In the first year post-transplant, between 5 and 14% of patients in those trials had their randomized therapy of either belatacept or the extended release tacrolimus discontinued due to adverse events. So a that not an insignificant percent of patients needed to discontinue their randomized therapy due to adverse events. Also, if you look with more granularity at some of the data from those studies, in the lower left-hand corner, all of these the regimens that were used in these studies included MMF. And if you look at the Astrograph registration data in the far left, in terms of the MMF doses, you can see that by 3 to 6 months or 91 to 180 days post-transplant, about half of the patients in that trial were receiving less than the protocol-defined and labeled dose of MMF being 2 grams a day. So 49 to 51% are receiving lower than 2 grams a day, and those dose reductions are due to adverse events. And if you look on the tacrolimus side, going back to some of our older data where we were trying to define the appropriate concentration range of trough levels of tacrolimus, one of the endpoints that we used was toxicity or an adverse event that required a dose reduction. And this was in the first 6 weeks post-kidney transplant. And you can see in the low group, in the lower right-hand corner, a third of patients required a dose reduction of tacrolimus in the first 6 weeks post-transplant due to adverse events or toxicity of the regimen. Therefore, you can see that a very high percent of patients have their immunosuppressive regimen, be it tacrolimus, MMF, or MMF, decreased due to adverse events early or at least within the first year post-transplant. So let me move then from the hard outcomes of death and graft loss to 2 other reasons why we should be focusing on safety events and safety endpoints. The second is that the impact it has on patients, as well as the incidence of the adverse events is high, and that gives us the ability to actually substantially show superiority and improve on those adverse events in clinical trials. To look at these 2 aspects, I pulled the most common adverse events that I saw in those 3 pivotal trials from the 3 immunosuppressants that were most recently approved for de novo immunosuppression after kidney transplant. And what you can see is that the highest or consistently highest adverse event actually is diarrhea. And common across these 3 studies and these 3 regimens were the inclusion of MMF into that regimen. So no doubt that type of adverse event, although it may not impact death and graft failure, is very impactful to the patients who are experiencing that. And you can see other most common adverse events, things like hypertension and diabetes, that clearly have an impact on the risk of death with a functioning graft due to cardiac disease, as well as the function of the graft long term. And then finally, the most common serious infection, the most common adverse event that's serious is the infectious complications that you can see in about a quarter of patients. And all of these are within the first year post-transplant. So as you can appreciate, adverse events not only impact patients, they are of high enough rate that they should be a concern for the risk of death and graft loss and the contribution they would have to that. But we also have the ability, at least statistically, to improve upon these with reasonably sized studies in de novo kidney transplantation. The last reason that I would propose as a basis for focusing on safety events is that, as Peter Nickerson described, one of the approaches towards efficacy endpoints is efficacy non-inferiority. And when I was at Astellas and working in the pharmaceutical industry, we would hear from some of the executives at the company that it was difficult to develop new immunosuppressants in kidney transplant because we couldn't improve upon the efficacy rates because our current endpoint, that being efficacy failure driven by BPAR, is so low in incidence that we wouldn't be able to show improvement and therefore superiority of a new therapy. So even if that is the case, and hopefully tools like the Ibox will enable us to be able to show superiority of new immunosuppressants, but if we can't, from the efficacy standpoint, innovative therapies may be targeted to improve safety. And if we do that, we can stimulate investment and innovation in transplantation by being able to improve the safety of these therapies. So you may now ask yourself, since the adverse events, as I described, are already included in the current FDA-approved labels, can't companies already actively promote safety superiority? So why do we need to include safety endpoints or consider them for development of new immunosuppressants? Well, although those safety events or adverse events, adverse reactions are included in labels, if you'll look at the details, and it's not often that we look at the footnotes within tables of approved labeling, but as an example here, this is a footnote on one of the adverse reaction tables within the Envarsus XR label that states the study was not designed to support comparative claims of Envarsus XR compared to Crolimus immediate release, the control arm, for the adverse reactions reported in this table. So what that is telling you is that although that data is available on the label, it wasn't designed to support the comparative claim that there might be superiority in one of these adverse events with the new therapy over the control arm. And that's not unique at all to the Envarsus label. Looking at both the Nulogix and the Astrograph XL label, you can see the quotes, the first one being from the Nulogix table, that's virtually identical for the adverse reactions there in their two studies, being the benefit and benefit extend, as well as in the Astrograph XL table. So you can see that the statements about not designing the studies, the registration phase 3 trials to support comparative claims is a consistent statement in the labels of our most recently approved immunosuppressants. So that should then lead us to say, how do we design a clinical trial to support comparative claims so we don't have to have that statement in the label when it comes to key safety endpoints? I thought it was informative to look at the FDA guidance on multiple endpoints in clinical trials, and particularly focusing on secondary endpoints, because what I would propose is that these safety endpoints that we'll be discussing should be or could be key secondary endpoints within a de novo kidney transplant trial. So what the FDA says about those safety endpoints, a few important quotes I felt from their guidance are that the positive results on these secondary endpoints can be interpreted only if we first demonstrate a treatment effect on the primary endpoint family. So in other words, first demonstrating that the efficacy hurdle is reached. And that might be, again, in the non-inferiority scenario, or it could be in the superiority, but it doesn't preclude our ability to first show non-inferiority on efficacy and then test for a secondary endpoint for potential superiority and safety. Second important point is if the success on the secondary endpoints is important, the sample size to support that should be considered when designing the study. So we need to think about not only what it would take to reach our efficacy endpoint, but also to show the superiority in these safety endpoints. And as I showed in terms of overall incident rates, I think we're in a situation where that should not be a driver to make these studies prohibitively large. It should be practical to design trials to show superiority in safety endpoints. And the third important point is that the list of secondary endpoints should be short. And we have to appropriately consider adjustments for multiplicity, therefore be very cautious in terms of choosing the number of secondary endpoints. Sometimes you'll see protocols where there's an exhaustive list of secondary endpoints with no specificity and hierarchy. And it's very difficult then to appropriately correct for the multiplicity in the testing of all of those secondary endpoints. So with that in mind, I've tried to put together what I think is some of the important operational aspects of incorporating safety endpoints as important secondary endpoints within our de novo kidney transplant trials. So the first is, it's really important, it's vital to predefine The secondary safety endpoint may be singularly if there's one important one, or a very small number if there's more than one, or safety endpoints. So they should be predefined, not simply a result of data mining or post hoc analysis. Secondly, we need to collect these safety endpoints rigorously and systematically in all patients and make sure that's defined by protocol, and not in the typical, well, here's what we found in spontaneously reported adverse event fashion. Thirdly, it's important, I believe, as we start this journey in kidney transplant, to use endpoints that have been established from trials and approvals of other therapeutics. And I'll give some examples of those. That sets a precedent that I think will be important, and then we can hopefully move beyond that. And then lastly, perform hierarchical statistical testing, as I mentioned, after first testing for the primary efficacy endpoint, but build in the appropriate statistical approaches to control for multiplicity and the overall type 1 error rate. So where does that lead us in terms of safety endpoints in kidney transplantation? What I would like to propose is we have safety events and an associated endpoint that are already clearly established. And I will call these the first generation. I think that we should be able to include these in de novo kidney transplant studies now. And not in particular order, but I think the first one at the top is the primary driver, and that being diabetes. We know that diabetes is important not only for its contributions to patient death, as well as contributions to graft failure. And we have very solid endpoints that are objective, such as hemoglobin A1C. So that is an event that we can pre-specify now, that we can statistically test for, and we can collect in all patients in a very straightforward way in terms of the hemoglobin A1C. But there are other important events, particularly those that contribute to cardiac death and cardiac morbidity, such as hypertension, hyperlipidemia, which we should be able to collect appropriate objective data now. Infections, using endpoints like plasma CMV DNA, which has been used for the approval of a antiviral, CMV antiviral. Leukopenia, which we know is problematic, particularly with MMF-containing regimens as an example, where white counts with the differential is so important. Anemia, as I've shown, is one of the most common adverse events after kidney transplant, where we know hemoglobin has been used. And finally, weight gain or obesity in our patients, which contributes to morbidity and mortality, where body weight is a very straightforward measurement. So I would propose that these safety events with the associated endpoints shown here should be considered now for inclusion as key secondary endpoint or endpoints in de novo kidney trials. And where can we go from there? I'll call these the second generation. I think we are very close to being able to move beyond those that I've just shown on the previous screen to things like plasma BK viremia as a measure of infection risk, particularly as the precursor to the risk for BK nephropathy. For PTLD, which is a low event rate, particularly in the adult transplant population, potentially using plasma Epstein-Barr virus DNA as a monitoring tool. Diarrhea, I mentioned, is a very high incident rate adverse event, and one that impacts the quality of life of transplant recipients. And we know that stool count, as well as validated scales such as the Bristol Stool Scale, have been used for the approval of drugs in this area, not in transplantation, but in other areas where diarrhea was part of the endpoint. So we have good tools for those, for quantitative assessments. And then finally, insomnia. When you speak with transplant patients about adverse events that impact their life, insomnia is included in that. And we have, again, quantitative methodologies such as polysomnography that can be used as an endpoint theoretically in these trials, and has been used in the registration trials of drugs to treat insomnia. So with that, I'd like to close by indicating, I hope I've convinced everyone that safety endpoints in kidney transplant are important for us to discuss and to include. As I see it, these adverse events that were discussed are associated with the immunosuppressants, the standard of care that is currently used today, and contribute to death and graft loss long-term in our transplant recipients. We do have objective and quantitative endpoints, safety endpoints. These have been used for the approval of agents in cardiovascular disease, as well as in other areas, and could be and should be incorporated into our trials as well as into our promotional claims in the label if we're able to demonstrate superiority. But to do that requires really systematic prospective planning. And that has to include not only the biopharmaceutical sponsors, the investigators and academic community that are working on these studies, the patients so that we listen to their voice and what impacts them the most, and regulators, FDA, EMA, and other regulators around the world. That interaction prospectively before those trials are designed or at the time of design is really vital. But this has the potential of being a catalyst for bringing innovative new therapies in transplant immunosuppression into our field, so that simply the non-inferiority on efficacy is not the barrier, but superiority on safety can be the advantage and can facilitate the introduction of new transplant immunosuppression. I want to thank you for your attention today and look forward to the panel discussion, and thank the TTC for inviting me to make this presentation.
Hi, my name is Rebecca Speck, and I am going to speak with you today about the regulatory qualification procedures for the clinical outcome assessments. I am a clinical outcome assessment scientist at C-Path within our clinical outcome assessment program. So here's the outline for this presentation. I'll start with a brief introduction about our COA program, and then speak specifically to our patient-reported outcome consortium, and then move on to highlight patient-focused drug development and FDA's clinical outcome assessment qualification program, and then tie that together with speaking specifically to the essential properties that are necessary for fit-for-purpose clinical outcome assessments. So our clinical outcome assessment program at C-Path includes 3 different consortia: our patient-reported outcome consortium, our electronic clinical outcome assessment consortium, and a rare disease clinical outcome assessment consortium. And one of the key roles of our COA program is to collaborate with other C-Path programs or consortia seeking regulatory endorsement of clinical outcome assessments for use as endpoint measures in treatment trials. Our program has 6 scientific staff and 7 administrative or project management staff. So speaking a bit more about our patient-reported outcome consortium, because that's where all of the work we do for qualification occurs. So this consortium was formed in 2008 by C-Path in cooperation with FDA's Center for Drug Evaluation and Research. We have 26 members that are all pharmaceutical firms. And our additional participants that we work with include representatives from FDA, from the National Institutes of Health, as well as collaboration with clinical consultants, patients, academic researchers, and contract research organizations, all toward the goal of either developing new PRO measures or clinical outcome assessments, or gathering evidence that's necessary for the work we do for qualification. The overall mission of the PRO Consortium is to establish and maintain a collaborative framework with appropriate stakeholders for the qualification of PRO measures and other clinical outcome assessments that will be publicly available for use in clinical trials where COA-based endpoints are used to support product labeling claims. Some of our goals, which are important to highlight, include the pre-competitive collaboration that occurs within C-Path and having that immediate FDA input and their expertise at the table. Obtaining FDA qualification of PRO measures and COAs is obviously another key goal of this group, so that they are then publicly available for use as primary or secondary clinical trial endpoints. And through these goals, we are trying to avoid development of multiple endpoint measures for the same purpose. And because of the collaborative nature, we're able to share costs in developing these new endpoint measures or in going through the process that needs to be done to gather evidence to support qualification. So just to highlight some of the successes for this consortium, in total, 8 clinical outcome assessments have been qualified through FDA's COA qualification program. All of these have been patient-reported outcome measures, and C-Path has achieved qualification for 5 of these 8 measures that are listed here on the screen, the most recent of which was issued in December of 2020 for the Diary for Irritable Bowel Syndrome Symptoms, which has a focus on constipation. Currently, the Patient-Reported Outcome Consortium has 15 clinical outcome assessments in 9 different therapeutic areas within FDA's COA qualification program. These include 12 patient-reported outcome measures, 1 observer-reported outcome measure, 1 performance outcome measure, and then 1 that's based on activity monitor data. So why, have an interest in patient-focused drug development? Why do we do all the work that we do within the COA program? Well, part of that is motivated by what came out of the 21st Century Cures Act, which included a mandate that opened up opportunities for FDA and the field of patient-centric research. Mandate includes focus on patient experience data, patient-focused drug development guidance, streamlining patient input, and reporting on patient experience drug development. And just one of the things that's highlighted here on the screen is this focus on the fact that data are accurate and representative of the intended population, including methods to collect meaningful patient input throughout the drug development process. So legislation effective in June of 2017 required that all drug approvals include a brief statement summarizing any patient experience data that were submitted and reviewed as part of the application. And this data primarily falls into one of 2 buckets, or potentially both. One being data regarding the impact of disease or treatment on patients' lives, and the other data regarding patient preference about treatment. With this mandate also came a series of FDA guidance documents on patient-focused drug development. The first was finalized and released in June of 2020, which is on collecting comprehensive and representative input. The second, released in February of this year, is on methods to identify what is important to patients. The third, which came out for review at the end of June of this year and is open for public review until the end of September, is Guidance 3 on selecting, developing, or modifying fit-for-purpose clinical outcome assessments. And final guidance, Guidance 4, which is not yet available for review, it will be on incorporating clinical outcome assessments into endpoints for regulatory decision-making. So when it comes to assessing clinical trial outcomes, FDA uses primarily 2 clinical outcome assessments or 2 different types of clinical trial outcomes to evaluate efficacy, one of which is clinical outcome assessments. And that's assessing clinical benefit through how a patient feels, functions, or survives. This is going to include patient-reported outcome measures, observer-reported outcome measures, clinician-reported outcome measures, performance outcome measures, and then another type which really doesn't fit into any of the other 4 COAs is data that's collected passively via mobile sensor technology. So thinking about, as an example, actigraphy and perhaps having an interest in step counts, as an example. The other way to assess clinical trial outcomes is through biomarkers. so long as they have been, validated as surrogates representative of future clinical benefit from a patient's experience. So now to speak to the FDA Clinical Outcome Assessment Qualification Program. So the intent of this program is to expedite development of publicly available clinical outcome assessments that can be widely used in drug development. The definition for a COA to be qualified is FDA's determination upon review of evidence that the COA and proposed context of use can be relied upon to have a specific interpretation and application in drug development and regulatory review. Importantly, once qualified, clinical outcome assessments become publicly available to be used in any drug development program so long as it's for the qualified context of use. And one of the major benefits for sponsors when these qualifications occur is that additional evidence supporting the COA does not need to be submitted by sponsors when used in the qualified context of use. And at the bottom of your screen, you see a link to information about this qualification program and guidance regarding that. So a little bit more specifically about FDA's COA qualification program and the specific stages that are involved in the content that's required for each stage is shown on the screen. So with the letter of intent, it's expected that the concept of interest and the targeted context of use are well articulated, and also that there's a detailed description of the clinical outcome assessment in question, and that the unmet need of that clinical outcome assessment is well described and rationale provided. Now, if the letter of intent is accepted, move on to stage 2, which is assembling the qualification plan. So that's where you'll provide the qualitative evidence, which is going to not just include the results, but study protocol, the results themselves, any of the interview guides that went into the qualitative data collection. You also submit a quantitative statistical analysis plan. So this is for the psychometric evidence that needs to be provided for the performance of the measure. And along with that statistical analysis plan, the study protocol that goes into that data collection and any concurrent measures that are used in that study would also be submitted with the qualification plan. Assuming that is then accepted, and you can move on to stage 3, the full qualification plan. Then I'm going to resubmit all the materials that were from the qualification plan itself, and then adding in the results from the quantitative study that would have occurred and all those results that are going to demonstrate the psychometric properties of the measure. I'll also be submitting at that point the data sets and any data dictionaries that went into those analyses. So when it comes to review of these qualification plans, there's essential properties that are going to need to be demonstrated to establish that a clinical outcome assessment is fit for purpose. And this evidence is going to be qualitative and quantitative. On the screen here, we're looking at the qualitative evidence that's necessary. So first off, it needs to be demonstrated that the content of the measure is relevant. So what does that mean? It means that the measure is going to reflect experiences, perspectives, needs, and or priorities of patients. And largely what this boils down to is the impact of the disease or condition and its treatment on how the patient feels or functions. So this could include signs and symptoms of the disease or condition, could include chief complaints, so maybe the most bothersome signs or symptoms, and possibly the effect or the impact of the disease or condition on activities of daily living and functioning. The other primary piece of evidence that needs to be demonstrated is that the content of the COA measure in question is comprehensive. So is the measure inclusive of all key facets of the concept of interest? So let's say, for example, your concept of interest is upper extremity physical functioning. Well, does the measure include range of motion? Does it include flexibility? Does it include strength? Is it including all those key components that are relevant for upper extremity physical functioning? As I said, the other piece of evidence that's going to be needed is the quantitative component. So first part of that is, is the measure precise? And the two things we're thinking about here are construct validity and reliability. So with regard to construct validity, is it demonstrated that you're really measuring the concept you intend to measure? And that's looked at through a variety of ways. So first off would be, are the scores on the measure in question correlated to scores from other measures that are going to evaluate the same or similar concept? You need to demonstrate that they're correlated to a certain degree to have, support that you're on target with measuring the correct thing. Inversely from that, you would want to be able to demonstrate weak correlations with measures that are measuring unrelated concepts. And then another piece of information that would be needed is to be able to show that your scores differentiate among groups that to be different in that concept. So again, to go back to physical functioning, we have a measure for physical function. We would expect the scores to differ based on ECOG performance status, for example, of the patients. The other component of precision is reliability. So are we measuring the concept accurately? The first piece of evidence for that is internal consistency. So are the scores consistent across items that are measuring the same dimension or measuring the same concept? And then the other piece is test-retest reliability. So scores are consistent over time when things are stable. So for example, in week 1, somebody has 5 migraines, and in week 2 they have 5 migraines. Nothing's changed for them. They're stable. You would expect that their responses on a patient-reported outcome would then be stable too. Another component that's necessary for fit-for-purpose clinical outcome assessments is demonstrating the ability to detect change. So scores are sensitive to change under conditions in which change has happened. Another term for this is called responsiveness. So to go back to our example about migraine, say from week 1 to week 8, an individual's migraines decreased by 50%. So we know that their experience of their disease and condition has changed by 50%. You would also then expect them to have a change on their patient-reported outcome that's assessing the impact of migraines over that same period of time. And then finally, are the scores that you're seeing on these measures interpretable? So what do the scores mean? Is a high score good? Is a high score bad? How high is high? Do we know what the range is? Is it 0 to 100? Is it 0 to 60? What is the range of scores that are possible? Are there certain thresholds within that range of scores that have meaning? For example, do they indicate a certain degree of severity? If you know that a high score is bad and your score is going to range from 0 to 100, that's one thing. But if you have evidence that demonstrates very specifically that a score above 75 is associated with these certain important clinical things that indicate severity, that makes your certain threshold have value or have information associated with it. And then finally, is a meaningful change in that score established, or is there a meaningful difference in the score that's been established? So, or a certain amount of time, is there certain meaning for a score change of 4 points for an individual on a 0 to 10 rating scale of pain? Or is there certain meaning to mean score differences between 2 different groups? So that is the end of this presentation, and I thank you for the opportunity to be part of this workshop. If you have any questions following viewing this presentation, feel free to contact yourself. Thank you.
Hi, I'd like to thank the organizers the opportunity to present today. My name is Dave Tabor. I'm a pharmacist by training, health services researcher in transplantation, and I focus on medication safety and medication safety outcomes in transplantation. And I will be presenting on patient-reported outcome measures specifically in transplantation. I will refer to these as PROs or PROMs throughout the presentation. They're essentially synonymous the way I present them. I do want to qualify that I am not an expert in developing or validating these measures. Rather, I use them in clinical research, specifically related to medication safety and medication-related outcomes in transplantation. And so that is my area of focus, and that is the angle I am taking for today's presentation. So here's my outline. First, I'm going to discuss a fair amount of the time. I'll focus on the rationale for using PROMs in transplantation, and then what PROMs to consider using as we move forward, when to administer these PROMs, and how to administer them. I will not spend a lot of time on this because this is an area that is still evolving, and certainly the real focus is the rationale and what PROMs to use. And then where we currently are with PROM development in transplantation, as well as the next steps. So those of you who work in transplant have probably seen figures like this in almost every presentation. They're almost obligatory at this point. But they do set the stage for the rationale for the use of PROs or PROMs in transplantation. So the figure on the left has been adapted several times. This is an updated figure presented by Cooper and colleagues in a recent publication in CJSIM. And what it shows is on the x-axis is time over a few decades, and then on the y-axis is the 1-year acute rejection rates. And this is depicted in kidney transplantation, but as a lot of the data I speak about today is specific to kidney transplantation, but certainly mirrors outcomes in other organ transplants as well. So you can see since transplant's inception in the late 50s, early 60s, the acute rejection rates have been very important in documenting our successes. And you can see that early in the inception of transplant, rejection rates were very common. The majority of patients had at least one rejection episode. And then somewhere in the early 1990s, we began to develop more therapeutics. This is when transplantation really took hold as a gold standard treatment for end organ diseases. And you can see the development of a good number of highly efficacious agents, including cyclosporin, followed by tacrolimus, mycophenolate, and then induction agents such as thymoglobulin and basiliximab, followed by mycophenolic acid and sirolimus. And you can see the rejection rates went from about 50% to under 10%, and now they are roughly 5 to 8%. So we've been very successful in reducing the 1-year acute rejection rates in transplantation. Also from this screen, you can see, or from this figure rather, you can see that the number of new agents entered into the market specifically to prevent or treat acute rejection in transplant has markedly diminished. And really our most recent novel therapeutic agent to prevent rejection was belatacept, approved in 2011. So now we're going on 11 years without a new agent. And some of that is attributed to our previous successes. When your rejection rates are so low, it's hard to differentiate yourself as a potential viable therapeutic option compared to the gold standard regimens that are currently used. So our successes have been remarkable, but that has also led to our current situation where therapeutics are not necessarily a predominant focus within the industry. On the right side of this figure, you can see the opportunities we continue to have. And again, this is specific to kidney transplantation, but these trends hold true regardless of the organ type. They're actually probably even more important in thoracic organ transplant, lung and heart specifically. And what you can see is the graft attrition rates. These are the graft loss rates annualized based on time from transplant. So the blue line is 0 to 1 year, red is 1 to 3, 3 to 5, green and purple is 5 to 10, long-term outcomes. You can see we had remarkable successes in reducing the graft attrition rates over the past couple decades, specific to the early post-transplant period, which is that blue line. They went from 20%, so 1 in 5 graft losses in 1989, and now we're below 8%. We're approaching 5%, so that's 1 in 20. That's a remarkable improvement in survival. But what you also can notice from this figure is that the long-term outcomes, although improving, are not improving to the same degree. And in fact, the 5 to 10 year long-term graft survival has remained fairly stagnant over the past decade. So this certainly offers us opportunity to improve long-term outcomes in transplantation. So what is driving some of these disparities between improved short-term outcomes but stagnant long-term outcome successes? Well, certainly part of this is the medication utilization in transplantation. So as I showed you in that previous figure, immunosuppressant regimens have increasingly become more potent and certainly more complex, such that the norm currently in transplantation is a quadruple therapy, which includes an induction agent and 3 maintenance agents, typically tacrolimus, mycophenolate, and prednisone. On top of that, because these agents are so potent, our risk of opportunistic infections is significantly higher, potentially than it even used to be decades ago. And so we're extending anti-infective prophylaxis beyond several months into even 6 to 12 months post-transplant. And we typically use 2 to 3 agents here, an antiviral, an antifungal, and an antibacterial or anti-PJP agent. Because we're becoming more sophisticated in how we monitor patients, both with the drug levels, but also the biomarkers used, dose modifications of these agents and titrations are becoming more frequent. And that's also inducing more issues related to drug-drug and drug-disease interactions as these regimens become more complex in patients that are older with more comorbidities. As depicted in the figure on the right, we are also more aggressively treating comorbidities. And this is data from the PORT study, which was a prospective observational study in renal transplantation. It was multinational, it was large, and it had data follow-up for a long period. And what you can see is the use of these agents, particularly to treat or prevent cardiovascular-related diseases and mortality, have dramatically increased, such that the typical kidney transplant recipient discharged from our center and most centers in the country are on at least 13 agents. And this is, again, this is mirrored in other transplant populations as well, and even more so in heart and lung transplant recipients. Now, layered on top of this is the fact that we know from data, including the study here, that long-term outcomes are also suboptimal due to non-adherence, okay? And one of the main issues with non-adherence is that it's hard to detect, and if you do not detect it early, it can quickly escalate into acute rejection that leads to potentially graft loss and or death. So this was a landmark study by Solaris and colleagues. This was a prospective observational study, again, multi-center. What this did is enroll over 300 kidney transplant recipients that were undergoing for-cause biopsy. And they followed them from that biopsy until they graft loss, death, or the end of the study, which was roughly 3 years. And what they found were about just over 60 patients eventually had graft loss. And in 2/3 of those graft losses were directly attributable to acute rejection. And remarkably, almost half of those acute rejection episodes were directly linked to medication non-adherence. So if you do the math, that means 1 out of every 3 graft losses in this study could be attributable in some capacity to medication non-adherence. So why is medication non-adherence so common and so important in our long-term outcomes in transplantation? Well, it gets back to what I was discussing earlier, and that's related to the therapies being difficult to take and difficult to tolerate in our patients. So this was a study we did in our transplant center as a single center, prospective observational study, longitudinally following patients over time. And we wanted to assess 2 things. 1, medication, self-reported medication adherence, as reported by the patients, as well as medication tolerability, again, as reported by the patients. And to do so, we administered 2 validated surveys, the Mariski Medication Adherence Scale, which is copyrighted, and we did purchase copyright approval to use this, as well as the MTSOSD-59, which is a validated survey to measure side effect burden. And we enroll patients anytime post-transplant, and we ask them to do the survey cross-sectionally at the time of enrollment, and then to consent to longitudinal surveys every 2 months if they agreed, for a maximum of 3 years or 36 months after enrollment. This is not actually complete data. We're still doing the survey as we speak, but it is a substantial portion of the data. So in this, we have 268 transplant recipients that provided 472 assessments longitudinally. You can see the mean time from transplant to survey was 4 years, and we had kidney, liver, and heart and lung patients in this study. And some of the results were quite interesting. First, medication adherence was reported, high medication adherence was reported in about 3/4 of patients, which means 1/4 of patients actually self-reported low adherence, as defined by the Mariski scale. And you can see that in the bottom left figure that the medication adherence was actually lower in livers and heart and lungs versus kidneys, which was not something we expected. And you can also see the side effect burden was slightly higher in the heart and lung patients versus the kidneys, which is that the orange dots with the dotted line in the bottom left figure. In the top left figure, you can see that medication adherence actually declined after transplant, which is something we expected. So as time from transplant increased, the self-reported medication adherence, high medication adherence decreased. And then in the top right figure, you can see that side effect burden actually decreased as well across the population. So patients tended to report improved tolerability over time, but nonetheless, still fairly high side effect burden, even 6 years out from transplant. I think the most important component of this study is in the bottom right figure, which correlates medication adherence on the y-axis to side effect burden on the x-axis. So as you can see, as patients' side effect burden increased, their self-reported high adherence decreased, such that those patients that reported high medication side effect burden, which is in the top 50% of patients, had 50% lower odds of reporting medium to high adherence. And for every 1 point increase in side effect burden, there was a corresponding 1.3% decrease odds of reporting high medication adherence. This makes sense. clinically, and it's probably fairly intuitive, that as patients have reduced tolerability to medications, they're less likely to be highly adherent. I think the remarkable components of this is just how tightly these measures were correlated, and how just 1 point increase in tolerability or decrease in tolerability could explain a fair amount of the medication non-adherence that we saw in this study. So it's no longer simply about dialing down our acute rejection rates to the lowest possible level. Just as an example, as I showed you in that previous slide, cyclosporine, azathioprine, and prednisone, there were about 50% acute rejection rates. That means that 50% of patients actually did fine on this regimen. Yet with the more potent regimen of thymoglobulin induction, tacrolimus, mycophenolate, and prednisone, where the rejection rates are 5 to 8%, that simply means that we have to over-immunosuppress roughly 35 to 45% of patients to achieve those lower rejection rates. So we're exposing patients to a lot more potent immunosuppression, which does reduce acute rejection rates, but at the cost of over-immunosuppressing a large proportion of these patients. So to optimize outcomes, we should really begin to do better at focusing on the overall patient experience and the burden we're placing on transplant patients with these complex and potent immunosuppressant regimens. And that's one of the strongest rationales for using PROMs in transplantation. So just as an example, I want to quickly give diabetes after transplant a nod, because this is so important in our outcomes, in our quality of life, and it's a good example of how we can differentiate the immunosuppressant agents. So on the left, you can see this is a review article, pretty detailed, looking at outcomes associated with diabetes across all organ types. And basically, it shows that across all organs, except maybe heart transplants, but certainly in kidneys, livers, and lungs, diabetes does increase the risk of graft loss and particularly mortality. On the right side, you can see a study done by Dukes and colleagues that looked at quality of life using 3 validated measures, the EQ-5D, the Health Utility Index 3, and the SF6D, which is a composite of the SF36 and then specific to kidney transplant quality of life. And across all of these, consistently demonstrating that those that had diabetes, either controlled with a blood sugar less than 126 or greater, had significantly lower quality of life scores. So again, we know diabetes induces hardships in our patients, and we know it reduces their quality of life. And we also know from a fair amount of robust data that the risk of diabetes is intricately tied to the immunosuppressant agents we use. So the study on the left shows the risk of developing diabetes with tacrolimus and sirolimus compared to cyclosporine as the referent agent. And you can see that particularly for tacrolimus, and also sirolimus, the risk goes up over time, such that at 5 years after transplant, it's roughly twice the risk of developing diabetes as compared to cyclosporine. We also know that corticosteroids are a major risk factor for the development of diabetes, as demonstrated by this study from Woodall and colleagues on the right side. This was a multi-centered, randomized controlled trial in kidney transplant recipients comparing steroid withdrawal versus continued steroids. And there was a lot of outcomes presented, but probably the most clinically relevant and significant differences between the groups was the incidence of diabetes requiring insulin, as well as A1C control, all favoring the corticosteroid withdrawal group. So we know that diabetes is a major issue. We know that it impacts quality of life, and we know it's a differentiator for our immunosuppressant regimens. Yet in practice, it's rarely used as a consideration when designing immunosuppressant regimens. perhaps if PROMs were more prevalent in our clinical trial development, this would take more information to the clinician when designing these regimens. So moving on to what PROMs to measure. I think there's lots of them out there, and I have a slide on some of them that have been validated in transplant coming up. But globally, we should think of PROMs that are very important to our stakeholders, and we should put our patients and caregivers ahead of provider systems and regulators when we're designing what PROMs to use. They should serve as differentiators for our therapies, using them as alternative endpoints versus our traditional clinical outcome endpoints. And I use the term alternative as opposed to secondary endpoints, because sometimes secondary puts in a connotation that it's less important than the traditional, and I don't want to provide that connotation. So I call them alternative endpoints as differentiators. They should provide the global patient experience after transplant as best as it can be measured in the patients that are actually delivering that outcome. And they should not forget our most unique and vulnerable populations, which includes the pediatrics, the elderly, which are growing populations in transplantation, as well as the heart and lung recipients, those with reduced capacity to understand the regimens and health literacy, those with low social support, or in underserved communities. Those are particularly important as we begin to think about delivering PROMs within clinical trials. So what to measure? We have begun an AST endorsed plan to do a national survey of all transplant recipients on the unmet needs of immunosuppressant therapy. So to start this endeavor, we convened a group of 8 transplant professionals, and we identified 15 constructs that holistically measure the patient's post-transplant experience. After multiple discussions and talks, we then added 2 more, and then we ranked these. And we ranked them with the original 15, and then we provided these 17 to 22 transplant recipients, and you can see the breakdown there: 14 adults, 8 pediatrics, 13 heart, 5 liver, 4 liver, 5 kidney, 1 lung, and 1 pancreas, and you can see the ages there. And we had those patients rank these constructs in level of importance to them. In this figure here, depicts on the left side the 8 professionals ranking in order 1 to 15. The bottom 2 were added later, so those were not ranked by the 8 professionals. And then the 17 on the right side are how patients rank these. And a couple, there's a lot of information here. I don't want to go through this in any fine-combed detail, but I just want to point out a couple of interesting observations here. First, there was actually fairly strong congruence between the professionals and the patients in the ranking. I was interested to know that we ranked them pretty close, particularly the top 5 were ranked. There were a couple that were swapped, but they were ranked pretty close to even. Side effects were ranked number 1 by both. So again, medication tolerability. And then 3 quality of life measures, 2, 3, and 4 were in the top for both groups as well. These are things where the agents could affect their ability to participate in life activities, have a high quality of life, and then how it impacts their comorbidities, such as diabetes, as I mentioned. So again, consistency across the groups and areas that are important to patients and providers' perspectives. The last point I'll make is that the risk of or the concern about rejection was actually ranked 8th. And this is one we didn't rank because we added it later, the professionals. So again, I think it's important to note that quality of life and medication tolerability trump the risk of rejection, which is our historic measure of efficacy for these agents. So when and how to measure these, I'm not going to spend a lot of time on this. These are just some of my recommendations using these. Longitudinal data is better than cross-sectional. Obviously, it adds a layer of complexity in gathering longitudinal data, but with electronic means, it's actually quite feasible. You can send surveys via links in emails or text messages and embed those within mobile health apps so that they complete those surveys pretty easily. And a lot of clinical trials are actually doing this now. You should match the timing of key endpoints in the clinical trial to this data as well, if possible, and synchronize these validated measures to the frequency of time to prevent recall bias. So if it's asking about the last 1 month or 2 months, that's when these measures should be asked every couple months, as an example. And then where to start and where to go next. So I borrowed this figure from Bill Fitzsimmons, who put it together regarding the primary efficacy endpoints and the secondary or surrogate, as well as safety outcomes. Again, I focused on diabetes. And on the far right are the patient-reported outcomes that have been used in transplant. Some of these developed in transplant, others developed in other disease states, but then validated in transplant populations. So some of the validated instruments, there's overall quality of life, health-related quality of life, side effect symptom tolerability, such as the MTSOSD that I referred to earlier, as well as disease symptoms and GI distress and neurotoxicities, which are common issues in transplant patients. And then finally, the PETS, which is the patient experience and treatment burden. This has been developed. It's actually quite an interesting prom in that it asks across a lot of domains, and then it's actually got a supplemental specific to kidney transplantation that was just recently validated. And then future areas of opportunity include being able to better assess comorbidity burden, and then again, those high-risk populations that have been notoriously forgotten about in the development of these prompts because they're harder to study, frankly, pediatrics, thoracic, due to smaller numbers. And then, as I mentioned, doing more longitudinal assessments versus just the cross-sectional analyses. So to summarize, I think there's very strong rationale to use PROMs or PROs as important alternative endpoints in clinical trials, and they can certainly serve as mechanisms to differentiate our therapeutic options in transplantation. Side effects and quality of life, treatment burden, and life experiences are key pieces of information that patients want us to know that that's important to them after transplant. Risk of rejection, although important, is ranked much lower by patients. Numerous PROMs have been developed and validated in transplant. We don't have to reinvent the wheel. We can use measures that have been developed in the general population and validate them in transplant. And as I mentioned, there's a lot of areas of opportunity, including intentionally measure PROMs as key alternative endpoints to allow for this differentiation of therapeutics, which will maybe help allow clinicians to better design therapies without agents that are currently approved, or spur the development of new agents as mechanisms to differentiate them from the current therapies that are clearly efficacious, but also clearly toxic and induce hardships on our patients. And so with that, I'll end, and I'll look forward to the discussion and the question session that's coming up at the end of these presentations. Thank you for your time.
Hi, you can't see me, but I'm here. Can you hear me?
Yes.
Great, because if not, Nicole and Amanda were going to kill me for not logging on 45 minutes ahead of time. Even though I've been probably kicked off, kicked off the internet about 10 times during this talk. Great talks, and I want to take this couple of minutes just to thank Nicole and Amanda and Luke, and especially Bill, who joined TTC, how important I think this TTC initiative has become, and actually how important it is for us to continue, because I think the talks today outline many different paths we could take, but especially I think, shoring up the real world evidence piece, which wasn't evident when we first started, that would be so important. Peter, there was a question from the audience about the treatment of the first rejection episodes and whether you took that into consideration in what happened to those people developing a second rejection episode.
Yeah, so we didn't control for it in the way that they're asking, other than the fact that we treated almost the majority with steroids. There was only a few that we used thymo as the initial treatment. So the numbers weren't there to really make a difference in that sort of control type of analysis.
I think all of your are sometimes, I think there's another sort of a thread that goes through, and this is the enrichment strategies that you're really talking about, right? Whether it's a de novo or whether it is an intervention trial later on. And I wonder, Peter, if you could hypothesize that maybe the Ibox score might actually, since it's already going to get some sort of approval somewhere, we hope, might be actually a endpoint for an intervention trial for, let's say, rejection.
It potentially could be, Mark. I think the worry I get with any kind of use of Ibox is I don't want it to be a surrogate, right? I want it to be I think we need real hard endpoints that we could use. Sure. If it's a surrogate, you're going to get stuck with doing the trial to the clinical endpoint, right? And so I think that's somewhat of a downside. So in a treatment trial, I think as long as whatever we're using is considered the hard endpoint, what you'd approve a drug on, that's what I'd be looking for.
And we're going to run out of time, but I was going to ask Rebecca and Dave the same question, basically, and that there's a lot of different PRO measurements you could use, right? Are we going to end up with a composite score? Are we going to just look at one? Are we just going to look at diarrhea? What's the metric that at some point we have to all agree and then stop arguing over it and like we did with the Ibox and say we're going to support this.
So this is Bex. From my perspective, I think that needs to be informed by the patient voice and by the patient's experience and what they're, informing us as to what's most important to them. And from many of those measures that are currently used, they, as Dave pointed out, some of them are generically designed. They're used across a lot of different disease states. So I think it really needs to be driven by what's important most to patients.
Yeah, I agree totally. I think just a couple of extra points. One, some of it's going to be defined by the patients you're studying and then the regimens you're using. I think we have a pretty good sense of the major tolerability and toxic issues based on the regimen. I will also say there's a lot of push to use the PROMIS measures that are being developed by the NIH because they're being developed specifically for these reasons, and they're being done very well. And they're obviously NIH supported, so there's no copyright issues and things. So I would imagine we could land on some of those more global PROMIS measures, as kind of like a key library, and then pick from those based on what we're specifically trying to study.
And I'm going to have to cut the discussion short, and I just want to make a comment about Bill's presentation. I really loved it, and I agree with all the data that he presented, obviously. And I thank the speakers. I'm going to give it back to Amanda.
It's Ken. I think I got a text. We may have lost Amanda. But I would like to close the meeting without delaying, just to say that in transplant for the last 2 decades, transplant scientists, transplant clinicians really wanted biomarkers and surrogate endpoints, but we had no idea how to go about doing it. We thought a peer-reviewed paper was the same as a regulatory endorsement, or should be. And it's really a credit to C-Path and the FDA working together, showing the power of this public-private partnership to advance something where we as transplant professionals were stuck forever. So I want to really thank them for driving this initiative forward and for organizing such a great meeting. Oh.
Yes, and I wanted to add to this and really express my appreciation to this cross-functional group, which keeps driving towards making a difference in transplantation, not only with a focus on endpoints, but the endpoints in my mind will be really very instrumental in terms of reviving drug development and transplantation. We all know that drug development is at this point in a really lull in transplantation. We need new drugs, we need better tolerated drugs, and we can bring drug developers only back in if we have a way to measure potential improvements we can see with those therapies. And we have heard a lot of ideas today how we can incrementally improve on that. Obviously, having a qualified surrogate endpoint or qualified reasonably likely surrogate endpoint would be a very important first step. So we are really looking forward to the progress, and obviously many congratulations to C-Path for continuing to drive this forward. Thank you.
Okay, we are at time, but I want to thank everybody for joining and participating, and really the dedication for the TTC members and data contributors. This is a labor of love requiring the international transplant community, data sharing, expertise, resources to fund such endeavors. So thank you all for your time, and please send any emails our way if you have any follow-up questions. Bye.